Bapak-bapak dan ibu-ibu yang kami hormati, ladies and gentlemen, distinguished participants, thank you very much for coming to our event. This is our second international seminar on One Health, New Roads Towards One Health in Indonesia and the region through the Noself mechanism. I guess we are all ready to start. So may I welcome Professor Dr. Bungaran Saragih of Ecotalis, also our former Minister of Agriculture, to give us his welcome remarks. Professor Bungaran, may I have your time, please?
Selamat pagi. His Excellency, Minister of Health, or the one who represent him. He is supposed to be here, but he is now in the United States. The Honorable Chairman of National Research and Innovation Agency, Badan Riset and Inovasi Nasional BRIN, or the one who represent him, but maybe soon he will join us. Distinguished speakers, esteemed participants, dear colleagues. Horas. I bid you all a warm welcome to Bogor and the EPB Convention Center and our One Health seminar. I appreciate your presence, all the more because of the current risk that social mobility gives to exposure to the pandemic. Without doubt, today is a special occasion because I recall we had the first seminar on 28th of October 2019. This was just a few months before the outbreak of the current global pandemic. Our second seminar today takes place about 3 years into the pandemic. The pandemic challenged everyone in the world, both citizens as well as government, in their respective roles, choices, and option of response. The heart of the matter is obviously that with increased mobility, the risk of disease jumping from animal or plant to human is increasing. All have to learn how to live with the current situation with regard to health. Since we spoke about the benefit of the Noself mechanism back into 2019, the Noself mechanism has therefore assumed bigger proportion. The needs of country, including Indonesia and other countries in the region, have grown. And now are many. One very clear need concern a way forward toward health for human, for animal, plant, fish, water, and the wider environment. One Health is essential, and pressure especially from urban areas, and also tightening international and national supply chains and related industry will drive development. Even though globally huge public money were spent on vaccines development. Globally, there is also a shortage of resources devoted to sustainable and environmentally acceptable step forward. In most countries of the world, the response of the government have included the centralization of its allocation capacity to government-funded and public research. Indonesia is no exception, as the President Jokowi decree in 2019. National Research and Innovation Agency, or in Indonesia we call it Badan Riset Inovasi Nasional, BRIN, is to take the responsibility of allocation and use of government fund for research and innovation. I think that we can say that BRIN is here at the right time. Furthermore, we also notice how universities, especially in Indonesia and elsewhere in the region, are also concerned with the One Health concept. Some universities established a One Health collaboration center to involve in the study of this new paradigm. Let us be mindful that the collaboration among government institutions, research institutes, and universities will enable us to understand the complexity of the global health problem and effectively mitigate the possible causes. I personally understand that this forum is a blend of science, policy making, and business community. It should therefore be able to make use of our insight for the sake of a healthy society. Many roads will then involve the use of current and advanced methods such as molecular biology and bioecology in both agriculture, maritime, and health sectors. As a new developing approach, I understand there will be some dispute regarding potential use of it, particularly the so-called self-DNA. This forum will then could be the platform for all of us here to reach the same angle so that we could realize benefit of this new paradigm maximally. Esteemed participant and dear colleague, today we are honored by the presence of distinguished researcher from University Peredico Naples, Italy, Professor Mozzolini, will share general insight in the Noself mechanism. And we will also learn from Dr. Marcelo Diano about algae and its unique role in devising application. Dr. Rija Putranto will discuss application in the oil palm industry, the biggest industry in this country. And Dr. Romy Nofrianto will present possible applications in the marine and fishery sector. I'm also grateful to Dr. Joko Pamungkas for his willingness to speak about One Health in Indonesia as a whole. I would like to personally thank Mr. Peter James, CEO of A&D Biopharma, for his continued commitment to the developing world in his endeavor to share the benefit of the Noself mechanism in a wider context. I would like to express my personal and deep appreciation to him. I hope that soon Mr. Jens will be able to travel again. He is not here. So we can welcome him here in Indonesia and the region. In Asia in general, and definitely also Indonesia, there is not a great deal of investment in research and development in agribusiness and also in pharma, I believe. This paucity can be explained historically and also by the generally very high competitiveness of Asian factor markets. It is all very understandable. There are a great many of generic products, all more or less functional for the purpose they were designed for. So the status quo and business as usual continue. But there are many signs that the efficacy of previously efficient weed deciders, such as Roundup, the famous Roundup, reduced, while disease has sprung up for which no effective remedy has been found yet. Historically, it may be so that the period of easy costing is coming to an end, because there is now a growing need for investment by agribusiness and pharma alike. Investment is part of responsible business and also very much part of good and sustainable business. I wish all of you success. I understand that the seminar has been organized to maximize interaction so as to involve you all. And I would like to invite you all to use the opportunity for sharing information. We highly appreciate your presence, and I personally would like to extend my thanks to PT Ecotalis Technology Indonesia, who organized this event. I wish you all fruitful deliberations. Thank you.
Good morning, everyone. First of all, thank you for your coming to the second international seminar on One Health organized by PT Ecotalis Technology Indonesia. And we, or some of us, may be already here twice since 2019, before COVID. We conducted the first seminar on One Health. Thank you for your coming, and we will have a program for today, starting from this morning until this afternoon. And the organizer would feel sorry about this delay. We are about 30 minutes late. This is due to we arranged our VIP guests, and fortunately, they both will come and join with us here. So we will have 3 presentations this morning before refreshment, I guess. And starting from the initial, what you call it? Indonesia used to be called as a provocateur. This Peter Jen. He's from A&D Biopharm, but unfortunately he could not join us here. But he will present his speak with some video. Yeah. And then we'll continue with the presentation or a speech from the representative of Minister of Health. And very specifically, he said he apologized because he couldn't join this seminar because he's still in the US. And last night he told me he will send his staff here. And he will be represented by the acting head of the Agency for Health Development Policy. I don't know whether it's already here or still on the way. And it is actually she. Yeah. She will be here in a minute. And this is the new organization that used to be Agency for Research and Development for Health. Now, because they are joined with the BRIN, Agency for Research and Innovation, National Research and Innovation, then they convert to the new agency in the Ministry of Health. And then the second speech will be the representative of the head of National Research and Innovation Agency. Dr. Handoko, also apologized for not able to join with us here because they have a launching program. They call it INARI. INARI as Innovation of Research and Innovation this morning in Jakarta. He is going to be represented by the head of Research Organization for Health. And this is also she, right? Yeah. And then after that, we will have a short question and answer, and before morning refreshment break, if time permitted. If not, the refreshment will be served on the table, I guess. Yeah. Okay, and then in the next session, we will have our outstanding guest here from University of Naples, Professor Stefano Mussolini. And we also welcome him because it is the second time he came to this room, I guess. Yeah. He's going to present the update of the One Health, or one aspect of One Health, about the evidence of the prospect of Noself technology for combating pests and disease. And then next to him will be his colleagues, Marcello Diano. He's also coming from Italy. He's representing the M2M company. And he's going to share with us about the developing Noself technology product using microalgae as production and carrier platform. And then short question and answer, and we'll continue with lunch break at about noon for 1 hour. The afternoon session will be presented by Dr. Risa Ariputranto, DEA. Is from the Indonesian Oil Palm Research Institute, Bogor unit. He will share with us about the potential application of Noself DNA for agriculture in Indonesia. And then the next speaker for this afternoon is Dr. Romi Novriadi. He's from Polytechnical University of Fisheries, Ministry of Maritime and Fishery in Indonesia. He will share with us about the current finding on self-DNA for fishery in Indonesia. And the last speaker for this afternoon is Dr. Joko Pamungkas. He is from Indonesia, Bogor Agricultural University, and he is responsible for Indonesia One Health University Network, or Indohan, in this university. He will share with us about the reflection on the One Health concept for animal and human diseases, or zoonosis. And then after this presentation will be some general discussion, and networking will be led by our single author or single actors, Dr. Taku William Botema. And after that, the closing will be about 3:30 p.m. and I will be adjourned this afternoon for this seminar. I think that's the agenda for us today, and I have also a function as moderator here, so I will continue my function here, sitting in the front. If the representative from the Ministry of Health already here, please come up with us here, and also from BRIN, Ibu, silakan Bu di depan, and we'll start with the video of Peter Jan first. But please come to join with me here. Could we start the video from Peter Jan? Peter Jan is the CEO of E&D Biopharma.
Dear Excellencies, dear Chairman of the National Research Innovation Agency, dear Chairman, distinguished speakers, participants and colleagues, and of course also hi to Nick, my son, who represents the next generation of crazies. As Professor Bungaran said, I caught recently COVID when traveling through India, and I'm very sorry I can't attend and meet the many friends I discovered in Indonesia, as well as the new friends from Malaysia, also here in the audience. 5 years ago, I fell very ill with a persistent bacterial infection, and from then on, I really wanted to find alternatives to the antibiotics that did not seem to work anymore. And I now know that Professor Mazzoleni's DNA self-inhibition principle is just what the world is looking for when fighting AMR, or antimicrobial resistance. Also, the first meeting I had after my recovery, the first car drive I could undertake was to meet Pak Takel at a nice castle in the south of Holland. And from then on, I got totally addicted to Indonesia. Its clever people, the warm welcomes, the impressive nature, the industrious cities, and the enormous potential it has, and on its way to becoming the 5th economy in the world. But it was also your motto, unity in diversity, that resonates with Professor Mazzoleni's invention. An oscillation between self and non-self, between the individual wants and the societal needs, that made me really want to commit to Indonesia and set up with Pak Takel and his impressive team the first One Health seminar. And let's face it, the Republic of Indonesia, with its neighbors united in ASEAN, is uniquely placed to become a world leader with the One Health approach, and that is what attracts me to your great countries. A young, bright research population, outward-looking, quick to unlearn the existing paradigms, reappreciating agricultural and health traditions, embracing this new and elegant finding that while for an individual organism, DNA is important within the cells, but one outside a cell, DNA plays a role in population dynamics hitherto unknown. Simply said, when an organism, any living organism, ingests or otherwise gets in touch at an early growth phase with its own extracellular DNA, it will stop growing and eventually it will wither and die. Professor Mazzoleni will explain more in detail, and Dr. Diano will let you know the ins and outs of the applications later today. And also Dr. Roman Novriadi will present a series of important in vivo trials here done in Indonesia. This new technology will offer shrimps and other patients an alternative to antibiotics and may one day safeguard society in case of a bacterial pandemic. And not in the least, of course, the technology also offers farmers a fair chance of reducing pests and plagues in their crops, affordable and accessible. So after a rocky and winding road and one viral pandemic later, here we are again. We mourn the many that had to leave us, but we can deliver the living a fully de-risked project, sufficient proof of concepts in vitro and in vivo in both health and agriculture, and this second One Health seminar. With pleasure, I have noticed that, especially in the ASEAN countries, researchers are very interested to learn and to discard old paradigms when new evidence points at new directions. And that's also very true in Indonesia. People unlearn rapidly, while in the West, big names, entire faculties and established magazines refuse to unlearn and steadfastly defend old views, sometimes to an absurd level of stupidity and to the detriment of farmers and patients. There are two paradigms I like to stress here, two new ways of looking at things. The two are different but connected, and together they will create a perfect storm, resulting in hundreds of new products and applications. Products that can be produced here by local companies for local problems and within ASEAN countries offering new export opportunities. The first point of view is that one can only work successfully in health, food and farming if all research and regulatory silos are much better connected. Research disciplines that can be connected must be connected. One Health is the new paradigm through which policies will be shaped, budgets will be allocated, and, who knows, people will vote. It is great that the Indonesian government is breaking down already the barriers and helps very different disciplines to connect and interact. The other new paradigm is about genetics. We all have been trained in the modern synthesis, basically stating that DNA is the blueprint of life. Now, another way of thinking is emerging, called biological relativity by Dr. Dennis Noble, where DNA is seen only as an organ of the cell, in a way recording and adapting to external influences. So context and environments are becoming perhaps more important than just genes and sequences. This biological relativity sounds heretic, and in some in the audience will cringe. Rightfully so, as this paradigm shift will, in time, influence research budgets, reprioritize applied science efforts, and surely also increase the interest in biophysics, a field that brings novel technologies as well. So beware, a change is about to come. Indonesia, with its ASEAN neighbors, would do well to embrace this biological relativity as well. Since the first One Health seminar, a lot has happened. Our partner, Corporate Biological Systems, built, together with M2M Engineering, a very serious factory in Pekanbaru, producing an array of micro and macroorganisms. Mr. Jonathan Koppert, here, can tell you about the many solutions his company already offers here in Indonesia. The Noself technology has been tested by state-of-the-art Indonesian research organizations. You will see some examples here today. Also, the interest of both commercial and state-owned companies throughout the ASEAN region is high and robust. Today, you will get a lot of information, perhaps too much to digest in one go. My colleagues are here to answer questions, and what's more, the Italian company Noself SRL, established by Professor Mazzoleni, Dr. Diano, and several others, will incorporate this year in the ASEAN group of companies. In this way, we can cater best to the needs of both farmers and patients in this geographic area. We are open for business and for scientific cooperation. We expect many diverse, affordable, and accessible products entering these markets soon, for which we certainly need the help of visionary politicians, regulators, and authorities here present. I very much like to thank Pak Taco and his team for the impressive organizations of the seminar. I hope that more and more younger people will embrace our novel ideas and carve out a future for themselves in the best interest of the many. And a personal message to Professor Bungaran: I dearly miss our chats. Thank you.
Okay. Thank you, Peter. Hope you get well soon and can join with us for the next event for One Health promotion. Ladies and gentlemen, I should apologize because just got the message. The representative from Ministry of Health still in Batam. So we will miss this representative. Then I should move to Professor Dr. Niluh Putu Indi Termayanti, who already joined with me here as the head of Research Organization for Health, Agency for Research and Innovation, National Research and Innovation Indonesia, or BRIN. So I won't read the CV because we have run out of time. So just please, Bu, you got about 20 minutes to share what the policy in research and innovation that might be related to the One Health concept, because you are coming from the research organization who deal with health issues. The time is yours, please.
Sorry. Okay, good morning, ladies and gentlemen. Can you share my slide? Thank you, Mr. Moderator. I would like to present with topic research and innovation focusing on to mitigate and future emerging diseases. I'm chairman of Research Organization for Health in BRIN. As we know, the pandemic is still ongoing, so we really understand that has underscored the continued threat of newly emerging and re-emerging pathogens and a critical value of research and pandemic preparedness effort and a national resilience of health. We know, as we know, the emergence and re-emergence of infectious diseases continues to threaten of the health of the people worldwide. Okay. So what we can do for the pandemic preparedness plan for the next pandemics, for the future pandemics. So there are three goals we want to plan. For the first, for how to we can increase the research, so especially for the characterize the pathogen of a concern and the surveillance to identify threats before they emerge. And the second one is how we can shorten timelines between pathogens emergence to outbreak onset and authorization or approval the candidate diagnostic and medical countermeasures such as therapeutics and vaccines. And this is the third one is very important, how to eliminate existing gap in research infrastructure and technology and expand preclinical and clinical testing capacity. The pandemic preparedness plan goal built on a foundation of the pathogen-specific research that include advancing research on priority pathogens known or having the potential for emerging as the public health threats. We have, for the pandemic preparedness plan, we have the epidemiology, infrastructure, technology, and coordination. So, if you can see on the slide, we have many experience with the disease. The outbreak have the 5 stakes before they can transmit it between human to human transmission. So, for example, for rabies, this on the in the stake 2, so only the disease only infected in human if the human can infected by bit by a dog or cat that infected by rabies. And the other hand, for example, for HIV, the disease on the stick 4, so no needed animal against. The disease can transmitted between human and human transmission. Okay, as we know, 70%, more than least 70%, the emerging or re-emerging diseases is zoonotic origin. So the zoonotic hydronating, the disease has emerged from an animal and crossed the species barrier to infect the human. Emerging and re-emerging diseases, we know are responsible for a significant proportion of the infectious diseases outbreaks that have the plague humanity over the age. For the impact, what the impact of the emerging and re-emerging diseases? Mostly we have the 3 impact, not only for the global mortality and morbidity. We know the impact. The second one is the economic burden, and the third one is the social and geopolitical implication. On the slide you can see, I think it's not very clear in here, how the economic burden, economic impact of the selected infectious disease during the outbreak. Yes. Okay. How to preparedness for the next outbreak or next pandemic caused due to zoonotic viruses or for the emerging or re-emerging diseases? For the best way for future preparedness for the zoonotic viruses, and to stop the appearance and reappearance of zoonotic, need to understand how we can understand for first time the primary host. Sorry. The primary host, and we must know research related the host range, and then what's the virus spillover event, and the frequency of the infecting the one host to another, then what's the kind of the immediate host. This is the framework there are 6 steps how we can do for the future preparedness for a pandemic. Step 1 is engage all stakeholders, and then human and animal surveillance systems, and then increase regional clinical capacity, and the next step in the step 5 is establish multidisciplinary data management and analysis times and analysis teams, and then engage government and international stakeholder. So the framework for how we can leverage our capacity and capability to zoonotic disease surveillance, especially in remote rural areas, that will facilitate integration into existing surveillance system. Especially for the pandemic or outbreak caused by virus, I think vaccine development for emerging infectious diseases is very important. So, in how we have many experience how do we implement using the One Health approach. For example, in the H5N1 outbreak in Indonesia, we have the nice collaboration with many stakeholders to control of the disease. We work together with the university, with the Ministry of Health, with the Agriculture Ministry, and another ministry. Okay. So, how to mitigate the risk associated with this yet unknown pathogen? The study will prioritize on the preparatory research on a prototype pathogen, and which are select pathogen identified from viral families known to infect human. And through the target basic and applied research on the prototype pathogens from each viral family of the concern, the accrual of solid foundation of the knowledge and medical countermeasures, MCM, such as therapeutic, vaccine, monoclonal antibodies, will enable with the rapid response when a novel pathogen from any of the viral families of concerns. What the continue the preparedness of the response, the preparedness research effort will provide the region roadmap of the product development and evaluation scientific infrastructure and study capacity needed to robust research respond to future pandemic. So, obtaining in-depth knowledge and development MCM for the prototype pathogen within viral families that pose the highest risk for the epidemic or pandemic. So the preparedness for major infectious disease, as we know, will save lives, and rapid deployment of the effective diagnostic vaccine and treatment can contain an outbreak before it's expand to a larger pandemic or outbreaks. And especially for the priority, how to priority and pathotype pathogen research. So we refer to this priority pathogen, and this pathogen specific research priorities viruses are most likely to cause a significant human morbidity and mortality. This one, how the research will conduct. We first time do pathogen selection, and then and to continue to basic the preclinical translational research, and the next to clinical research, especially for the drug and vaccine or therapeutic. The pandemic preparedness plan will support critical studies to how to characterize a prototype and a priority pathogen, including understand the viral biology and structure, host immune response, mechanism of immune evasion, disease pathogens, and studies to develop assay and animal studies. And fundamental knowledge of pathogen biology, structural properties, mechanism of transmission, including how we can identify the viral vector, viral life cycle, viral entry mechanism, and host receptor, tissue tropism, and host immune response is critical to effort informing the development of medical countermeasure and against new pre-emerging or emerging pathogen. So we what the BRIN can support to conduct, especially to health of regions, national health of regions. So we BRIN have the research organization for health. We have the 7 research center. So research center for biomedical, center of biomedical research, research center for preclinical and clinical medicine, research center for public health and nutrition, research center for pharmaceutical ingredients and traditional medicine, research center for vaccine and drug and Ekman, research center for microbiology, and research center for veterinary science. And the other hand, BRIN provide for the all research through Indonesia, provide the research funding, infrastructure. The infrastructure belong to BRIN can share with all the scientists through to Indonesia. And we have the human resources. This is research scope in our research center for biomedical research. So if we hope we can make the collaborate with the other scientists in the nation or in the Indonesia or from the board. This one, the research scope under the research center for biomedical about the diagnostic research, stem cell research, and phenotypic, genotypic research related to biofilm, IMR, micro and microbiology, and how do we conduct the population research, especially for genome-wide association studies and laboratory-based disease. And we have the research center for preclinical and clinical medicine. The research center conduct the research related to preclinical and clinical medicine, especially for communicable disease, and then preclinical and clinical medicine of the non-communicable disease and mental health. And the next, we have the Research Center for Public Health and Nutrition. So the research scope is very broad, So including the one maybe as we have the factor infectious disease and zoonosis in a human. We have the research group that the research group conduct the research with the topic of factor infectious disease and zoonosis in human. And we have the Research Center for Pharmaceutical Ingredient and Traditional Medicine. The research scope regarding the research of on the development medicinal raw materials, research on the standardization of traditional medicine with using the raw materials, especially from the origin from the Asia, and research from the development of traditional medicine, research on the formulation, and something like that. For the Research Center for Vaccine and Drug, we have 3 focus on our research. First of all, for vaccine development research, especially for we develop the protein platform, RNA and DNA platform. Halal vaccine is it's our focus in our research, and the biological product development research, especially we development of biopharmaceutical products such as the monoclonal antibodies, nanobodies, recombinant protein, and something like that, and drug research, how we can discovery of drug for the communicable and non-communicable diseases from the nature, from the marine, and something like that. Ekman Research Center for Molecular Biology, they conducting the genome diversity, molecular bacteriology, malaria vector resistance and malaria pathogen, dengue, hepatitis, RBC, cytogenesis, molecular therapy and structural biology, and stem cell and organoid cell. This one, the Research Center for Veterinary Science, the scope, the research activities are related the early detection, early report, early response for the animal and zoonotic diseases, emerging and re-emerging disease and zoonosis, bioterrorism, especially to biological risk, implementation of One Health, utilization of veterinary innovation, animal welfare, and Okay, we have the 5 focus research in our research organization. We have the one we call the program home program in our research center. First, the topic 1 related the drug and vaccine control of the infectious and regenerative disease, and the second topics, development of the research and innovation in precision and regenerative medicine, and and another topic with the development, how we can develop the medical devices and health protection tool, and the other topic we have the infectious disease research and innovation, and research and innovation for stunting reduction through a family approach. This is in our vaccine roadmap research in our research organization. We developed the TB vaccine, HPV vaccine, and then we developed the biological product such as the NH1 gene from the dengue and something like that. This scheme, how to BRIN can support in vaccine development in Indonesia. So BRIN provide the funding, especially for the in Indonesia, the lab scale, how to vector expression, how we make the master seed vaccine, and till how we production with the CGMP, and then preclinical testing. The funding, the BRIN provide the funding till the preclinical and clinical testing for how that one you can see in the slide, how the BRIN can support in vaccine development. This is for the infrastructure. So now BRIN nearly complete for the animal BSL-3, especially for macaca. So BRIN support by Pak Imung to develop how we develop and build animal BSL-3 for the macaca, for the preclinical testing for the vaccine and drug, and how we can prepare to mass scale for the vaccine. And BRIN have the genomic laboratory for all the working related the DNA, RNA, and something like that. And BRIN have the CGMP lab consists 2 lines, especially for CGMP yeast and CGMP bacteria. So for the structure and function studies, BRIN have the in cryo-EM in BRIN is coming in December 2022 at the genomic lab. So the infrastructure, the equipment can the equipment can share with another researcher through Indonesia to support the technology, including X-ray, crystallography, nuclear magnetic resonance, imaging, cryogenic electron microscopy, and something like that. And I will skip this slide. And in preclinical research, we conduct how to provide the expansion of the specialist technology related the high throughput cell sorting, cytometry, immunologic assay, sequencing, and support the genetic examination of the zoonotic reservoir that might contribute to emerging infectious disease of the global health importance. I will skip this one. So institution, including our institution, BRAIN, should leverage its expertise, especially in infectious disease, genomics, proteomics, bioinformatics, and access to clinical samples to develop a rapid response diagnostic for biological threats and emerging infectious disease. And especially for the therapeutic, we support basic translational and clinical research effort to identify promising target for intervention and generate novel therapeutics that are both pathogen specific and have a broad activity. For example, we development of small molecules such as the monoclonal antibody and have the provided scientists with the valuable tools to prevent and treat infectious diseases. One especially for the infrastructure, BRAIN have the many infrastructure there with another researcher, especially for the preclinical and clinical research infrastructure for the animal BSL-3, animal BSL-4 maybe, but we have we have no yet the BSL-4 capacity. For the clinical and preclinical research, preparators includes leveraging and expanding existing domestic and international outpatient and inpatient clinical trial sites, enhancing on-site expertise and developing the clinical research infrastructure needed to evaluate countermeasures and rapidly respond to infectious disease, especially disease. If we continue to clinical study.
Please your time, Bu.
Okay.
2 more minutes.
Okay. I think it's my last slide. So we need the effective coordination and communication are central to effective preparedness research effort, and requires a close collaboration and intramural and extramural programs as harmonized and supported by effective mobilization on logistical, administrative, and technical resource. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Ibu Profesor Niluh Putu Indi Darmayanti. Professor Indi, yeah, because in Bali used to be abbreviated by NLP. Yeah, NLP. Niluh Putu, yeah. Okay. Thank you very much for a very thorough presentation and related to the research and innovation policy in Indonesia. And this is something really new because the agency, the BRIN or BRIN, is close to 1 year, right? Yeah, almost 1 year. Yeah. Well, first, okay, thank you. Ladies and gentlemen, we have heard together how the complexity of health problem in the world, and it's come to the conclusion that One Health is one of the solution to how handling this problem. And Professor Indi also, on her presentation also mentioned about how the importance of the development of therapeutic and vaccine in conjunction with health remedy. And I think we will hear this more from our next speaker, but we are now having about 15 minutes for question and answer, or I think that if we will put together with the second speaker, because we will manage the time as the representative from Ministry of Health is not clear yet. So I would like to invite Professor Stefano Mussolini to come with us here. He's a professor from the University of Naples, Federico II in Italy. Go ahead, Stefano. You got also 20 minutes.
It's already on. Okay. So, thanks very much for. I'm really honored to be here for the second time, and I wish to thank the organizer for inviting me. Professor, really like to be here again. For addressing a welcome speech for the second time. I'm really like to be here again. I prepared a presentation. Can I have my presentation? Sorry, find it. So I like to start with a very, very general view that shows you my former background is ecology, plant ecology, and I like to show you how I arrived to this discovery and to understand the possibility of applying in the way that I showed you before, okay? So let's start with a very, very simple concept summarized by this statement. DNA is everywhere. What do I mean with this? If we move to a tropical forest, you have big biodiversity, lots of different trees, species, animals, microbes, insects, butterflies, everything is here, okay? And if we move forest, and you can actually think to go on a single leaf and to see a little lizard that is on that one, and the same leaves can have a cricket or a fungus growing on that leaf, you can imagine that each of these organisms has its own DNA, the self-DNA of each species. That's an obvious concept, okay? And you can imagine in a big forest how many different self-DNA for every species are there. But then the organic cycle, the life cycle, all the organisms also die, and they will fall on the forest floor, and they will start decomposition, and their DNA is still there, although if they are dead. And they start decomposing, and their content, the DNA that was in their living cells, is going to be released into the environment, into the organic matter that is in the soil floor, in this case. If you zoom in and you look at the microbiome that is in the soil, all the microbes that are there, these microbes are actually living in the water, in the organic matter, and are surrounded by all the DNA that is coming from all the organisms that are in that system. But the same microorganisms have a very high turnover. They reproduce and die. And every time a cell is dying, it's releasing its content into the system. And what is interesting to specify in this very moment is that when the decomposition is going on, actually the DNA molecules are the most resistant to the decomposition processes. So they stay in the soil in this case, also when all the other organic material compounds have disappeared and have been digested and recycled. And I say to students, smiling this, saying, think of those movies where the investigators find the killer DNA after many years. You know, it's the same concept. The DNA is very resistant. It can stay for a long time in the environment, okay? Now, what is interesting to understand is that all this environmental DNA is part of the nutrition of all living organisms. So plants, while absorbing nutrients and water from the soil and things, they also absorb the DNA that is present in the soil, the free circulating DNA. And this happens also to animals. When we eat food, okay, you have a pasta dish, I'm Italian, so I like pasta, you will have DNA of the tomato and of the wheat and of the olive that you use for cooking it, okay? So DNA is everywhere. And if you think of the trophic chain, all the animals and plants are then connected, and they exchange this DNA through the trophic network. So in the view that I'm presenting, when you look at a complex system like a forest, you have also to remind that in that system, the life cycles is circulating DNA everywhere. Go back to the forest view then. It should be clear now that there is a big system dynamic going on, flows of everything. I'm just saying, remember that it's not just flows of water and nutrients, but also of DNA. We should not be scared by this concept. DNA is a very natural molecule part of this life cycle. But what happens when we transform the landscape to agriculture? We start removing the natural vegetation. The land becomes very different. And when a big intensive agriculture system is applied, then you have one species only in substitution of the original highly biodiverse ecosystem. Then think about the accumulation of DNA in this case. And in this case, the DNA will be coming from only one dominant species, okay? So the view in terms of DNA circulation is going to be different. In a natural system, there is that network, very complex network of trophic exchange of DNA through all the organisms. In this case, there will be an accumulation of the self-DNA of the crop. And we are aware that if you, especially in dry conditions, you repeat the cultivation of monospecific crops for many years, then the productivity is going to decline. In Europe, we are very well aware that there is the so-called soil sickness that is not just a nutrient depletion. If you fertilize with chemical NPK fields that have been cultivated for too many years by the same crop, you will not solve the problem. And the ancient Romans, they more than 2,000 years ago, already wrote in their text about agriculture that rotation concept between crops was a good concept. In fact, when you have rotation between different crops in agriculture, in traditional agriculture, then of course there is not accumulation of the self-DNA of one crop, because you will move the crop in different places in the field, okay? So expert farmers know that it's a good practice in traditional agriculture. What is very interesting to mention, being in Indonesia actually, is that also rice paddy is an interesting case to understand, because with this type of cultivation, with the flooded by water, you can keep the monospecific growth forever. So you can have rice paddy that can be hundreds of years repeated every time without problem. Then why? Because DNA is soluble in water, is washed out, and so in this type of system, you don't have the accumulation of the self-DNA of one species only. You have the water flow that clean up the system continuously. So what I'm saying, this is a new view of this problem, is that in dry land, you have sustainability only with biodiversity. If you get monospecific crop in dry land, you face a problem. When you move to water system like rice paddy, you can not worry about, okay? That is sustainable also if it is monospecific. Now, let's come to no-self concepts. With this background, what I showed you before was my background, studies in agriculture and organic matter cycles and thinking of this DNA concept, I came up to this concept. Okay, let's have litter then, organic matter. You decompose it, and at the end of the decomposition, you have the humus, and everybody knows, also people that are not expert in agriculture, that humus is good for plants. So the plants can grow on the decomposed organic matter material, okay? We studied a lot this. I'm not going to give you the details on this. We did the chemical composition of fresh leaves and decomposed leaves, okay? And it changes, of course, because there is a lot of processes, biochemical changes going on during the decomposition. But what is the very important point is that the fresh materials, sorry, what happened? I'm saying that the fresh materials are not so good as nutrients for plants. The humus, the decomposed material, are good for plants. But if you think of microorganisms, the microbiome, it's the opposite. The fresh material is good food for the microbes that like to decompose it, and the decomposed material, the humus, is stabilized, stay in the soil, is nutrient for the plants, but not good anymore for the microbiome. So you have this concept that fresh material can be phytotoxic, stabilized material is a good fertilizer. This seems a very, very general concept. But I got this idea more than 10 years ago. I told you this also last time that I was here, that through the decomposition, it's true that you create humus that is a fertilizer, the blue arrows, but you also release the DNA. And if that DNA is the self-DNA of that species, then that can be inhibitory. And now we arrive to the point. Forget about that. We studied funny plants like these little plants dying in the middle or the regeneration in the forest, but these are details. If you want to discuss more, I can explain you more. And now let's stay with the story at a simpler level. The discovery was this one. If we put plants in presence of heterologous DNA of different species, they grow very well. If you put the plants in the presence of their self-DNA, they are inhibited, no germination and no growth. Okay? That's a very simple experiment, can be done by everybody, on any species. It works always on any species. If you check on bacteria, the same. And you do it on protozoa, the same. The self-DNA is inhibitory. And if you do it on algae, we observed with Marcello Diano the same effect. And fungi, the spore germination is inhibited by the self-DNA, but not by the DNA of the other species, okay? And in insects, sorry, there is a quite impressive. If a larvae of an insect is eating DNA from a food for it, there is a complete metamorphosis and completion of the cycle. But if the same larvae is eating itself DNA, the cycle is stopped, is inhibited. And now this concept is quite amazing. We start working then on model organisms like Arabidopsis in plants, and we start publishing things. It's very interesting. The root of the plant, in presence of the self-DNA, does not absorb it. The fluorescence that you see here is the DNA. It remains outside, and the plant becomes impermeable. It's not efficient in nutrient uptaking, and inside the cell, you don't see evidence of DNA uptake. Whereas, if you put in the plant non-self DNA, you see the fluorescence going inside because the plant is uptaking the non-self DNA that is a good food, and inside the cell, you see a lot of metabolic activity that is stimulated by that. So it's a very, very clear evidence on plant, on Arabidopsis plant. But then we start working on nematodes. We collaborate with the CNR Institute, the National Research Institute in Italy, and we get very clear results of very good life of the worms in the presence of non-self DNA and inhibition of egg and larval arrest in very significant results in presence of self-DNA. Ongoing results on Drosophila melanogaster, just to show you that the experiments now are very robust and tested on many, many different species. And very clear results. You can see by eyes, even if I don't make any comment on that. So, let's make the story very simple. All organisms in presence of an environment rich of non-self DNA, of DNA of other species, they are happy, okay? They are healthy, let's put it this way. But if you enrich the environment with their own DNA, then they become unhealthy, they are weakened. So this is the concept, what I call the no-self concept. And now we go to more in depth on the subject of this conference. If you think of the traditional phytopharmaceutical research, you start from any sources, you extract molecules, yes? Okay? And, or you can have molecules to be tested by chemical synthesis, that's okay. And you have thousands of molecules that can be searched and tested by random screening to search for their bioactivity and possibility of using them as drugs. But through this process, you arrive to have actually an approved drug in a long time. This process can take more than 15 years of research, and there are problems. Some of these molecules can be, the process is very long and expensive. There is not specificity of actions, and these molecules can be toxic not just for the pathogen, but also for others. Like if you use antibiotic, okay, the antibiotic is not good for the health of all organisms, not just for the microbes that we want to kill. And most problems comes from the appearance of resistance problem. Because in this case, if the resistance appears, this drug cannot be used anymore, and all the investment is lost. So we come up with this idea, very simple. Forget about this. It's a bit strong to say like that. And eliminate all the problems and just take the DNA of the pathogen and use that DNA as the inhibiting product. And we say that this can be the solution. 1 year only to do all the process, and a huge advantage in terms of R&D development, time and cost. Now, if the concept is clear, imagine a new future for agriculture. The traditional approach, if you have a pathogen here, this worm, the black worms, killing the crop, we spread synthetic pesticide. It's true that we kill the worms, but we also have residuals in the crop by the chemicals, and we also create damage to the ecosystem around the agriculture, okay? In our concept, we change the concept. We take the DNA of the pathogen and use it as the inhibitory product, and we get a healthy ecosystem with the plants that are very healthy in this condition. And in medicine, imagine that you have antibiotic treatment of microbes that is effective. Okay, the guy is happy because he's recovering. But if the pathogen is antibiotic resistant, then there is a problem because the antibiotic don't work. And we say, okay, let's test if the self-DNA of those pathogens can be used and cure the patient. So it's the same concept. And now we come to the interesting part of the story. Okay, already what I said was interesting, but that all organisms have a microbiome, okay? It's funny to see a plant compared to a man, but a plant is a living organism that uptakes nutrients from the soil using the digestion, the decomposition of organic matter that the microbiome does in the soil. It's similar to what happens in an animal. The only difference is that in animals and men, the microbiome is inside the gut. But still, it decomposes, digests the organic matter that is the food. Okay, it happens inside, but it's the same concept. So digestion is similar to decomposition in a soil. And why I'm saying this concept? I show you a very, very nice example that was told to me by an organic farmer that runs a very, very nice farm. He's crazy about organic agriculture. He would never add a chemical in his farm, okay? And he told us this story. His cows are fed with trifolium hay, very high quality, that he produces. And he takes the manure of his cow to fertilize the tomato and gets very good tomato. Then he uses the same manure to fertilize the wheat, and he gets very good wheat and makes wonderful bread. But then he used the same manure to fertilize the trifolium hay, and he got a problem, the soil sickness. Now, with my explanation, this is clear why. The DNA of this is going through the gut of the cow, is handled by the microbiome that is in the gut of the cow, and in the manure, this is enriched by the DNA of the trifolium. And so, this DNA is part of the nutrient cycle of these other crops, but it becomes negative, self-toxic to the trifolium. So, this is an anecdotal story. It's not science. It was told to me by a farmer, but it represents the way we went on, okay? We started working on this concept, and now I'm going to closer to the conclusion. Imagine then, in human medicine, you get a gut, and in the gut you can have a healthy gut, high biodiversity. Lots of bacillus, lactobacillus, good organism in our gut, making digestion of all the food that we eat. And we know that if our food is diverse, it's good for health. But if you get in a dysbiotic state, and you get a high level, for example, of E. coli in your belly, then it's unhealthy conditions, and this can be treated with this concept, DNA extraction of this red pathogen, and using it, provided by a natural library of yeast, for example, and then you bring back the dysbiosis to the balanced diversity of the microbiome. So it's the same concept again in a different situation. And now we come to the bridge for the next communication that Marcello will give. So we thought, okay, the concept is very powerful. We got an idea, using the DNA to control a pathogen, the self-DNA of that pathogen to control a pathogen. But how do we produce that? How do we distribute that DNA in the soil, in the environment, in the food, okay, according to the conditions? And so we start thinking, okay, let's use algae, and Marcello will tell you much more about that later, and the algae are known to be biostimulant on the plant. If you give algae in the water, in the irrigation, the plants will grow very well, okay? But then, let's give to the algae the DNA of the same plant. The algae will grow very well, identical to the other case, because they will add that DNA that is non-self for them to their uptake, but then the same algae becomes inhibitory on the same plant. The only difference between these two productions has been that this stage, they were exposed to natural decomposition of this plant material. So this microbiome, these algae, became carriers of the DNA of the target species. And now we come to the upscaling concept, and Peter Jens mentioned that with Koppert we already started a production in Pekabaru doing algae and upscaling them and things like that. So you can imagine a pathogen given to an algae, in some, sorry, not the pathogen given, the DNA of the pathogen given to the algae, and then you grow the algae, upscale them in the big facilities, and then you get this fertilizer product. But that fertilizer product is special because it will be dual use. It will be fertilizer for the plants and controller for the pathogen. So a dual use microalgae. And now we go to the end of the story, and I will show you a last example on oil palm case here in Indonesia. This is the traditional view. Organisms need nutrients. That's true for plants, for animals, for humans, okay? But then there are the pathogens, and we want to kill the pathogens. That's the traditional view. Every time there is a pathogen, we want to kill it, okay? That's understandable. We add this concept. We must be aware that the health of the organism depends on the possible accumulation of its self-DNA in its environment. And the weakness of the organism can enhance the pathogen. This is the Noself model idea, okay? We can change this by adding microalgae to the cycle that are inhibiting the pathogen and biostimulating the organism at the same time. Now I show you, and we think that this is really a One Health approach. Okay, but I will tell you a little bit more on this before concluding. We did, starting since the last time I was here in Bogor, some work on the oil palm case, and we simply demonstrated in the lab a very simple concept that now you should follow me because I told you already several times. If you add to oil palm the self-DNA of the palm, you can get inhibition of the growth of the palm, okay? Because the palm doesn't like to grow with its own self-DNA. But also, if you take Ganoderma and you grow Ganoderma in a plate, in a petri dishes, if you add the self-DNA of Ganoderma, you can observe inhibition of Ganoderma. So I'm repeating the same concept. The self-DNA is inhibitory on the same species. And now what's happened? I took this picture 3 days ago in Pekabaru. Oil palm plantation, thousands of hectares, monospecific DNA accumulation. The palms in first cycle of plantation have no problem. Second cycle, so-and-so. Third cycle and fourth cycle, they are very much affected by Ganoderma. So the old plantation that are more than 30 years repetition of crops, they start to have the Ganoderma problem, okay? And with all the things that I've been telling you, what do you think now of the practice of adding the empty fruit branches around a single tree to provide organic fertilization? That's a good attempt by the farmer. But in my view, you are creating a high concentration of self-DNA by the decomposition process for the palm. That's not good for the health of the palm. In the long run, of course, not immediately. Immediately you can get an organic fertilization. But by the accumulation of the self-DNA of the palm, you create a negative condition. And in that negative condition, you get the Ganoderma attack that can kill the plant. So in summary, it's just the last few slides, what we are saying that here is a palm, the decomposition of the residuals, the litter of the empty fruit branch of the palm is releasing the self-DNA of the palm, and if this becomes too much, This is creating inhibition on the palm root system and facilitate the entrance of the pathogen attack. And the Ganoderma then becomes happy and growing in that conditions. And remember the microbiome concept. What happened to all the microbiome that is in the soil? If the soil becomes full of self-DNA of the palm, the microbiome will be also an additional transmitter of the self-DNA of the palm. So it is a vicious circle that creates negative condition for the palms. And now, if we take the DNA of Ganoderma and create a natural library of algae containing the DNA of the Ganoderma, then this product can be grown and becomes a dual-use product that is a biostimulant for the palm and an inhibitory for Ganoderma. Simple like that. Sorry, I was wrong direction. Okay, I repeat the concept here. So it's, I think it's, so the conclusions. I've been explaining you now an example, the oil palm Ganoderma model. But in the way I'm presenting it, you should understand that then this could be described for the banana fusarium model. Banana is a big, extensive crops in the tropics, monospecific crops. After many years of cultivation, they're facing very strong attack by fusarium that is becoming a pest. And they're trying to compensate this with searching for resistant banana varieties, okay? But in my view, the problem is the accumulation of self-DNA of the banana that is enhancing the fusarium attack. So we can say the same story that I told you for Ganoderma and palms for that. But then, potato Phytophthora, tomato Robanche, we work in Italy on this. Grapes and Peronospora. So we can say this for any crop and any pathogen. It doesn't make any difference. Any livestock and any pathogen of a livestock, a fish, a parasite in the gut of the fish. We are doing research with the veterinary school in Italy by adding a food integrator to the fish diet, algae, with the self-DNA of the pathogen of the fish. And so we get stimulation of the fish and inhibition of the pathogen. That's the concept. It's always the same concept. And human, why not? Of course, humans take more difficult legal issues, but we are working on some institutes in Lisbon and in Napoli. Also. Okay. And so these are just examples of things. And our motto then is, as nature does, because we think that our approach is not adding a chemical by synthesis, by industry production things to the system, but is really to modulate the system in a natural way, as it actually does naturally, okay? And the last point, and then of course, if you have any question, I'll be happy to answer, is then when you spray our product, let's say that the algae with the self-DNA of a pathogen, then the day after, of course, you don't see anything, okay? It's not like spraying an herbicide and then the day after you see all the weeds killed, because that's a killer. In our system, nothing happened apparently, but starts a different dynamic, and then in 15, 20 days, the plant starts to feel a little bit healthier, and then the leaves becomes a little bit greener, and the pathogens becomes a little bit less aggressive, and then in some months, the system is rebalanced and becomes healthier. So that's why we feel that our Noself concept is very much a One Health concept, because it's not entering in a... yes, I finished. That's, I was last. My smile. Thanks, David.
Okay, make a big applause to Stefano Mussolini. Actually, I let you over time because it's very important, the subject. So, but still, we have another speaker next to you. But if you don't mind, later you can join here when we have a discussion, okay? Ladies and gentlemen, we have been shown together by Professor Stefano Mussolini how the impact of the One Health approach to mitigate what the complexity in the health problem that we face today or the future. And one very important thing that I note from this presentation is that a monospecific crop in the long term will not be sustainable. And this is very, very serious issues that we need to take care of. Because, for example, we all knew Indonesia is the first oil palm producing country, and this is handled with the monoculture, okay? But it is something that you mentioned, the old traditional technology like rotation and also organic input may also alleviate the impact of this self-DNA effect. This is very challenging and important, but still a question there where we could apply this agent to the target. And this will be the subject of Marcelo Diano. Please come with us here about how to develop Noself technology product using a microalgae as production and carrier platform. Time, just, okay, you got also 20 minutes, Marcelo.
Hello everyone, nice to meet you. I'm Marcello Diano from Italy, CEO of end-to-end engineering in Italy. Many thanks for participating and for coming. Thanks for coming to all colleagues, and many thanks also to Ungarn and Mr. Tago, Minister, Head of Research Agency. Very happy to be here again for second time, first time in 2019. So, we continue to discuss the evolution of history. How use macroalgae as a technique for production and carry as a platform for Noself technology. Thanks for Professor Mazzoleni to very, very interesting, presentation. So I continue the history. Just a brief introduction of myself and our company, M2M. I'm CEO. M2M Engineering is involved in scientific research and industrial application of macroalgae since 2000. So it's more than 22 years. Working in macroalgae with several customer collaboration in several countries, in Italy, in Europe, Sweden, Europe, and also in Netherlands, Europe, and also in other countries in Indonesia. I'm very happy to collaborate. We are making a serious collaboration with Coppert and Biocycle. Also, thanks to Mr. Tago to organizing this meeting, to Budi Cycle and Coppert. Thanks also to Jonathan Coppert to serious industrial production. This street and serious collaborative production in Indonesia for these developments. Street and serious collaboration industrial production. Indonesia for these developments. We have also several patents in macroalgae cultivation, above all to increase production, to increase production and optimize the productivity and have a increased production in industrial farms. Our experience, of course, we have several field of application, like in pharma, in livestock, in agriculture, also the other research and developments in the other new. So just a brief overview of our activities. We follow the entire chain from the laboratory strain maintenance. We have also our own strains. So strain system for maintenance, strain, laboratory for research development, producing very strict controlled production. Also industrial production, industrial photoreactor for production, open field, and of course all system and all the industrial production needs a strict monitoring and control system to control the whole production. And we are also applying to improve the production, improve the productivity, the biological modeling of macroalgae and interfacing with also the sensoring and monitoring system, also to increase production, maintain full control of the production to achieve the maximum productivity in all places. Several, as I said before, we have several collaboration around many countries, also in Indonesia. We are very happy with these collaborations. Peter Jens, many thanks to Peter to organizing and to push on this type of concept. As I agree with Peter, I very like the Indonesian environment, Indonesian people, because it's a very kindly, open mind, open to new innovation. So I think several of you already we meet and discuss in last years. So just very try to be short. General view the macroalgae. In general, we can view as four groups of macroalgae: green macroalgae, red macroalgae, diatom, and cyanobacteria. In general, macroalgae is a unicellular organism in order of the microns dimension that using sunlight and nutrients and CO2 from culture, using for growing, so also for production. And difference from seaweeds from the sea, it's an algae from algae with a leaf. So multicellular is like similar to the plankton growing on the sea, but differently, macroalgae is a unicellular organism that growing from the laboratory to so the order, the magnitude of each cell is the order of microns, and the type of chain of the production is different because seaweeds growing on the sea differently. Macroalgae is industrial production with controlled water, controlled nutrient, controlled system, closed system. So the type of approach in production and also harvesting In seaweeds, it's becoming from the sea, so also in harvesting, it's different type of the harvesting, some system or some also some manually harvesting. Microalgae instead is different. The type of cultivation is with the industrial production and also type of harvesting is industrial in controlled condition. So we continuously have under control the full production also in type of the harvesting. Type of system used for growing macroalgae can be photobioreactor is a closed system, can be a different type of system. These are some of our plants, right? Like a multi-flat or tubular, cylindrical, the other type of tubulars, and also the multi-flat and flat system. Or the other system can be open, like a thin layer residue. We prefer use for increased productivity and for some reason, to use the production open system, also in open system, but in controlled environment. So in general, we use this type of system, closed, or this type of system, also if are the open system like a raceway or a delay, but in controlled and closed greenhouse, so the controlled environment. For some reason, first about contamination, about controlled product, because from rain, from environment, it's a vehicle also of pollutants and some contamination. So in the end, the end product that we produce is very stable and very clean and controlled, certificate, and with the several strict procedure for also for control. So also in open system we grow in closed system also to maintain light under control, temperature air under control, and temperature of cultivation under control. Of course, one of the other parameters that we maintain with the strict control is water. We use clean water with the controlled system to also purify and to maintain control. So just summarize the different type of approach in seaweeds. It's in open sea, so we have also the variability of the end product, and also, of course, type of product is different because the one with the problem well known in seaweeds is also the contamination from heavy metals, for example, coming from the sea, because on the sea you cannot maintain control of water. One of the important achievements in our product is, for example, the contents of heavy metals. We have 2 orders of magnitude lower than seaweeds, but it's normal because the type of the industrial production is different, because we use a clean water and control it, and the maximum of heavy metals, it's, for example, it's 0.1, but many times, and also I'm very happy in the last analysis, many times we have undetected contamination, undetected heavy metals. The other important point is stability also, stability of product. In industrial controlled production, with the full nutrients under control, light under control, temperature of water under control. So in the end, during the full year, we have all batch, all product certified certified with the stable product, with the same characteristic, with the full parameter under control. This is very, very important for also for market access, for human, for application, for human, also for livestock, also for agriculture. Maintain under control fully parameters, fully contamination, maintain in stable range, and have always consistency and certified condition. A certified end product is important for also for many type of field of application, industrial product, like in agriculture, like in pharma, like also in livestock. The current applications in health food for human and also in pharmaceutical is also know this type of application. It's already commercial and also legal. What I want to also show the new innovative application that we are studying in the last at least 8 years. We developed a product for agriculture and livestock that they want to show the interesting things. We developed for agriculture a biosimilar product for seed care application, foliar and drip irrigation. I tried to very short. These are, for example, some picture of course, in the last 8 years, we followed the research and development laboratory to maximize the characteristics, to maximize the biostimulant effect, and to achieve the stable product and industrial product for agriculture. So 8 years, it's we made a lot of things in the laboratory, in greenhouse, controlled greenhouse, uncontrolled greenhouse, and also open field trials. Now we are going also the 4 years of the field trials, field trials. This, for example, some effect on seed care application in soy, in rice, for example. This is interesting things, for example, in application in problematic seed. Sometimes you have, some lots or some condition, some dormant seed or problematic seed. In this condition, this is an example of the very interesting effect of the stimulation coming from chemicals, from the hormones and the compounds and contents in the body. Please, next. Okay. So just as I said, we have also several trials ongoing, and also in the last 4 years we made a lot of field trials. This is a classical effect, for example, this in seed application wheat on several hectares field application. This, for example, in baby leaf, in spinach, in baby leaf in arugula. These are field trials for officially field trials for also for registration of product. So we are already in a commercial phase. This is an example of the big field trials randomized with the randomized plot on seed and foliar products. Next. This is the other field trials in several hectares within southern Italy. We are making a trials in several places in Italy, in southern Italy and northern Italy, also in several other countries. I also like to visit sometimes to start with trials. Next. This is the other official trials with the randomized plot, big trials on corn. This on tomato. Again, official trials for to have also a statistically for product for commercial. Tomato. Next. Also, we are applying industrial seed company for seed treatment industrial chain. So we are already in this phase, also in application in big industry in seed. Other trials in grapes, in Italy, grapes it's also very important. In grapes, table grapes and wine grapes. Next. Okay, but some just some numbers. The effect is increased yield. For example, we made also several trials in comparison with the other commercial product. For example, in this case. It's a test with three competitors, three products with the foliar application in wheat, and this is our product biostimulant. So it's also confirmed many times this type of behavior, this type of results on it. The other field trials comparison with the other seed care product. These two competitors. This is our product seed treatment application. Next. I want to just as I said before, the important thing in this type of product that we have a constant and consistency results. The product is very stable, but becoming from the type of industrial production, we have a whole batch controlled with the same characteristic, with the same efficacy in the end. We don't have a variability on product. Some numbers, some increase of yield in tomato, in lettuce, this in drip irrigation, this one in foliar application. So, coming to the livestock product, as you saw before in agriculture, the product, the macroalgae product, our product for algae, is a biostimulant product. In livestock, we have we are making we made and we are also going to the other trials with shrimps and fish and also the other animal to apply as health feed supplement. So integrating the indirect from the 1 to 5% of the total diet, if you add the macroalgae is in this percentage, you have in general enhances disease resistance and improve the health in animals. So, next please. So, as I said, we made and we are making we have a lot of ongoing things on this with the University of Naples, Department of Medicine and Veterinary, and also with the, I thank to Dr. Romy, also in Indonesia, on shrimps. Many thanks for the trials that you are doing. So in general, the effect on chicken, on fish, on shrimp, it's health improve of livestock. So increase stress and disease resistance, and this is a good effect that you can use also to reduce the antibiotics. The other big matter on health of livestock. Have the immunostimulant activity, better livestock health, and increase survival rate. This is very important because, for example, in hatcheries, in hatcheries, the survival rate in the first stage is very important in production because if you have a lower survival rate, of course you have a lower number of the shrimps to grow, and in the end the production will decrease. So, becoming to the yeah, slide.
3 minutes.
Yeah, okay.
Okay, good.
Come to the Noself technology. Imagine this macroalgae. I like this this example. As already showed Professor Mazzoleni, coming to the Noself technology and fragments of 17 years of Professor Mazzoleni. Imagine macroalgae is a truck. It's a truck with the biostimulant effect in agriculture, in plant, and also in livestock. Image is a truck. And image, if in natural way we charge a fragments on this truck, and we use this truck as a production and carrier for bringing agriculture for plant, or for bringing human as a carrier for example, we need some pathogens, or use this to for livestock to improve the health and decrease the pathogen in livestock. So the concept is, as a natural combination as nature does, image the macroalgae grow in medium and substrate with the fragments becoming from the discovery of the Professor. Give time to macroalgae uptake to these fragments. In the end, you have the truck charged with this fragment. So after this, image use this type of technology that I showed before, this type already industrial, ready for commercial, ready for production. Imagine this type of technology to produce a carrier for livestock, carrier for human, or for agriculture. Next, please. So, last year we worked a lot on these trucks, these fragments charged on trucks. We are already in field trials stage on some for example, in this case, it's a problem on fusarium attached on lettuce. These results on Noself technology with the macroalgae carrier, natural carrier on fusarium on lettuce. In the right, you saw the lettuce attached from fusarium. This is a problem. It's a very, very big problem because you lost also more than 50–60% of the total production. So, in this case, there is macroalgae macroalgae with as a natural carrier with the fusarium fragments from Western Amazonian technology. These are the results. I think is self-explained because the effect is dual. Is a stimulant effect, as you saw before, as the drug is a stimulant. So you have a stimulant effect and also inhibition effect on fusarium. So you have a good effect, stimulate the health and decrease the pathogen with this technology. The other, we have many things going on, research developments, some in first stages, some other like fusarium or like some weeds in tomato already field trial test. So we have a lot of things ongoing using this technology as industrial production and industrial final product for macroalgae with this Noself technology. So I think I finished. Thank you very much.
Give applause to Marcelo very much. And I would like to have you stay here, Marcelo. Marcelo, please stay. And I would like to invite Stefano also here. Unfortunately, Professor Milo already left. Okay. Ladies and gentlemen, we'll have a very short question and answer if you have one. As the latest, we have here from Marcelo that the commercial production of microalgae is available right now. So this is also some opportunity. We are in the tropical area with a lot of sunshine, a lot of water, but we haven't have any success production in such product like that. And this is coming the technology that developed by M2M from Italy. Well, let's have some participant to have a question, please. Identify yourself and to whom the question is towarded, and please make it short because we will entering the lunch break already. Thank you. Okay, the gentleman on my left here.
Thank you, Mr. Moderator. I have a question to Professor Stefano. There's a very interesting presentation, Prof. And I have two questions for you. First question is, do you think that we need a specific amount of the self-DNA to work well? I mean, to inhibit the growth of this pathogen. And the second one, I don't know whether you already used this concept to explore about the self-RNA instead of the self-DNA. Thank you.
Sorry, I didn't get the second one.
The second one, I don't know whether you already used this concept, go to the RNA instead of the DNA. Thank you.
The first one?
Go ahead.
Yes.
The first question is whether we need specific amount of the self-DNA to inhibit the growth of the pathogen.
Yeah. Well, in vitro, we observed effects with higher concentration than the one that we observed in the field. That was counterintuitive because in a petri dish, for example, on plants, we could see an effect of inhibition with 200 parts per million of concentration. But when we used it in the field, we observed the effect also with much lower concentration. And the understanding of this strange result was because of the microbiome amplification effect that I sort of try to explain. We are studying very much now the process of how the microbes actually uptake the DNA and eventually can amplify it. So we can provide as a product lower amount and actually observe higher effect by the interaction with the natural microbiome processes. This is under investigation. About the second one, on DNA and RNA differences. RNA is very labile, whereas DNA is very resistant. So, in principle, you can use RNA, for example, it is used for RNA interference approach, targeting specific genes, but then it must be protected the RNA model somehow, otherwise it degrades, it doesn't get the target, doesn't reach the target. With our DNA approach, it's a very robust technique because the DNA is going to be there and has a shelf life in the stock, in the farm for a long time, but also in the soil. So it's an easier tool to use.
Okay, this answers the question. If you're not satisfied, you get the minimum of the last 3 minutes. Okay, thank you. The second gentleman over there, I saw the one. Okay, go ahead.
Okay, thank you, Pak. My question is, how did the self-DNA work? Is it work for family or genus? For the example, we applied to same graminae, for example, rice, or to another organism. Is it still work for self-DNA? And second question is, I curious about the consideration to using of microalgae. Why you not use the fungi or bacteria with the same ability to improve plant health? Yeah. Thank you.
I understood the second
Maybe what? Or Stefano, yeah.
If you ask them if we can use other organisms to do the effects, yes, that's the answer is yes. Is not only the algae. We use the algae because we have all the know-how, the technological know-how how to produce them and upscaling industrial level. But of course, we are also working on other carriers, potential carriers that could be fungi or bacteria. And in that case, the upscaling should be done with different technologies. I don't know if this was the question.
Okay. Is there anyone?
Thank you for to give some inhibition action of DNA. It should be complete of DNA or particular part of DNA, because we will have a problem for production of whole DNA in a large amount in industry. Thank you.
It's a very good question. The basic technology is based on the whole genome DNA that is extracted from the target proteins, but then is fragmented in random fragments. The size that is relatively small, between 100 and 10,000 base pairs. So it's a big mix of fragments of different sizes and randomly broken by supplication or by other processes. Or by the decomposition in a natural way. When you decompose the organic matter, the result is the same mix of fragments randomly fragmented. So, in the traditional approach, we don't know which are the active fragments, if there are some fragments that are more active than others. Okay? But currently, we are now working on selection of some region of DNA and checking if there are variability of the inhibitory effect using this sub-portion. But this is more, how can I say, molecular biology application to improve the efficacy. But the efficacy is already very high using this very basic approach. Because we get, in the case of nematodes, 100% larval development stop in 2 or 3 generation nematodes. So when they are fed with their self-DNA, so it's a really very strong effect. Of course, we can keep working on improving the selection of DNA regions and making it more feasible.
If I can just issue on this. It's connected also to a question from Romy. In the real field, it's like a copy of nature. I like to displace, and the amount need, the effect is very high in field, and the quantity need, it's also very low in field. So already now, the amount, for example, used in field trials that I showed, it's a very low amount for hectare treatment, for example. We are saying about some hundreds of grams, so it's very, very small amount. So also also taking the full genome without any other things, it's very effective and the quantity need is very low. Is what I think.
Okay, okay, the last one, please.
Okay, thank you. So I have questions for Professor Stefano. So basically, for the self mechanism to happen, the plants or the organism have to be able to absorb the DNA. Is it correct? And after the organism absorb the DNA, I would like to understand what happens after that. Is the DNA integrate to the genome, or is it similar to the RNA vaccine, for example? I mean, is it going to be permanent, or is it going to be just for several months? And after that, I also have a question for Marcelo. I think I can understand about the inhibition happens by the self mechanism, but I am trying to understand what happens, how is the mechanism for the microalgae to be able to boost the growth of the plants. Is it just because we add the Noself DNA, or is it because of the microalgae can produce something that can boost the growth? For example, is it because the algae contain protein or something? Yeah, that's it. Thank you.
This is a very good question. Now, the question on how is the uptake mechanism? First, remember that the uptake is only for the non-self DNA. It's not for the self DNA. The self DNA does not enter the cells. It lives outside and it acts as an inhibition at the membrane layer of the, for example, of the roots or the cells. But when we have an algae, for example, uptaking the DNA, the non-self DNA, the DNA of pathogen, then this is probably incorporated into the genome. We say probably because it remains stable for many, many generations. The natural library that Marcello has prepared and has been tested, they can be grown in bioreactors for months. So going over many, many hundreds of generations of cycling, they are still inhibited. So they contain the self-DNA of the target that remains there. But when I say probably, it's because we are doing work now on long sequencing the DNA, searching for these fragments that And it's very difficult work for bioinformatics teams. Because what we understood so far is that the mechanism is not regarding genes, coding regions, but is somehow hidden in the non-coding part of the DNA. That is a massive portion of the genome, but is much more difficult to detect in terms of changes, because probably you know that it has been termed the junk DNA for many years, this non-coding part of the DNA. So somehow it's a totally new phenomenon that we are now trying to address, and we require help by molecular biologists, and biochemists, and organic chemists, and we are working with animal technologists to find these. We are now finding the first indications, but are still ongoing research. It's not a definitive question, okay? But it is stable. It stays there for a long, long time. Okay?
Before I closing the session, there is some question from me, Stefan. Some scientists doubt that the cell technology is basically is RNAi. RNAi. Is that true? And then, is there any other groups who's developed this technology in anywhere in the world?
Well, I don't know about the second part of the question. There are groups that I didn't understand what they
Well, maybe Europe and the United States or the other part who develop similar approach of this technology.
No, I wouldn't say so. Of course, there is a lot of work on RNA that is conceptually completely different. Because in that case, you use either cDNA or RNA protected. That is actually the same sequence of the gene that you want to silence. So the concept in that case is that you have a target gene, a specific gene, in the cell, in the body, or in the insect, and you deliver that RNA, or the copy of that RNA, and you obtain a silencing of that specific gene. But then, since you are acting against a single gene, it's very easy to get resistance in the treated population, because the population can adapt, and you can get individuals that are resistant to that action, and then you have lost your technology. In our case, since we are working with random fragments of a very broad spectrum of the region, the effect is not against a single gene, but is an overall silence of the whole metabolic activity of the cells. So we've done a lot of work in this direction. And for example, in the cells that are inhibited by cell DNA, you see a down expression, a down regulation expression of thousands of genes that go down. And the amount of RNA that is produced is very significantly reduced. So you really observe a wave of reduction of metabolic activity by the inhibition. In that sense, I feel quite confident to say that resistance cannot appear, because it's not going to be focused. So it's a very different approach from RNA interference.
Thank you. I think with this answer, we will adjourn our first morning session, and I will give back to Wuhan today, because it will be you after this. Then let's make a big applause to this our outstanding. Thank you very much.
Thank you, Professor. I think we've had such very good deliberations in the morning, and this adjournment calls us for some, I don't know whether it's DNA or food or whatever, but it's available outside.
Bismillahirrahmanirrahim. Ladies and gentlemen, welcome back to our afternoon session, and we will have three outstanding speakers. The first is Dr. Isha Hariputranto, DEA, and I would like to come forward, join with me here. He is the Vice Head of Indonesian Oil Palm Research Institute, Bogor Unit. I mean, located in Bogor Unit. We'll present the update on the proof of concept of the potential application of Noself DNA for agriculture in Indonesia. And then our second speaker this afternoon is Dr. Romi Novriandi. Is he here? Okay, please join us here. He's from Polytechnical University of Fisheries, Ministry of Maritime and Fisheries, Indonesia. He'll share the topic about current finding on Noself DNA for fishery in Indonesia. And our last speaker this afternoon is Dr. Joko Pamungkas from Indonesia One Health University Network, or Indohan. This is something new, and we'll be hear from him what this is actually this new institution in the university, Bogor Agricultural University. His topic will be reflection on the One Health concept for animal and human diseases or zoonosis. Ladies and gentlemen, we will have each of these speakers about 20-minute speech, and I would like all of them keep in time so we can have some time for discussion in the last session of this one. So let's start with Dr. Isha Hariputranto. Go ahead. Thank you.
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Thank you, Mr. Chairman, Professor Didi Ajar Gunadi, for introducing me. So maybe I would like to take some of your time, 20 minutes, because Taco gave me 20 minutes. So I will try to be brief and concise about what I'm going to present. I will introduce myself after this slide, but first I would like to let you know about the potential application of the Noself DNA technology for agriculture in Indonesia. But for disclaimer first, because in Indonesia this is still also a new thing, Professor Mazzoleni, we learned so much from your research and your team that you have done in Naples. So we would like to show you all a brief summary of what we have done in Bogor. Actually, we are a team of work. Myself and Galuh Pramatasari, and also Dinyastika Sari, Imam Bagus Nugroho, which he was no longer with us because he moved to other institution. Professor Didi Ajar Gunadi and Mr. Taco. Okay, allow me to introduce myself a little bit because basically I'm a scientist. So what I I'm going to present you is all about based on data that I have seen in my laboratory. So I have my expertise mostly on molecular biology, genomics, of course, working on a lot of DNA sequence in plants, in humans also. When I was in the Beijing Institute of Genomics, we learned so much about how to deal with the human genome that is really different with the genome from plants or even microbes. And during my PhD, I learned so much and studied a lot about the genome of rubber tree, so Hevea brasiliensis. So basically, I'm a laboratory guy looking for a sequence in front of my computer, a big screen normally, and try to understand the meaning behind every sequence of genome or even in the genetic aspect. So for the last 2 years, I worked a lot with many partners, and thankfully, TACO invited me. Thanks also to A&E Biopharma for giving us the opportunity, Professor Mazzolini also giving us the opportunity to learn and to study about the Noself DNA technology. Okay, so a quick reminder about what is the self-DNA mechanism. So at first, when TACO came to us and talked about this mechanism, because I was with Dini, TACO, you remember, I think you still maybe remind of our faces, right, when you talk about this mechanism, because I'm a molecular biologist, learning so much about DNA, the mechanism. Even we try to understand what is about the degradome, how to understand the sequence that already been destructed by the microRNA in plants. So when you talk with us about double-strand DNA that fragmented and give the what we call the effect to the plants or the organism that absorbed it's bubbling in my mind, because as a scientist we need to talk about the theory that should be proven by data. So in this mechanism that TACO once talked to me and give me the paper, he tried to show me this one, that self-DNA concept is that a inhibitory mechanism of DNA happen to the conspecific species. What is this conspecific species? Conspecific species is that the DNA came from its own organism. For example, if you want to inhibit yourself, being lazy, for example, you should take your own DNA and ingest it. This is only an analogy that maybe can make you understand better the concept of the self-DNA. Basically, when you have grass on your field, on your houses, and you mow the grass you will smell something like a weird planty smell. That's what we call allelopathy concept. Actually, this is a volatile organic compounds that went out from the plants, goes into the grass maybe 200 or 300 meters after, and the grass over there can understand. This is some type of communication between the plants. That grass will understand that this part of area has something wrong. For example, if the grass has been attacked by a pathogen. So they told the grass, the next door, to enhance yourself. Maybe do some fortification on your cell wall, or just enhance your self-development so that you cannot waste the energy or you can absorb or do something like developing a rhizome so that you can protect yourself. That kind of communication happens and already known for many years, what we call allelopathy. But self-DNA concept is perhaps for some people quite difficult to accept because you need to prove it, right? You need to prove it. How they say that the DNA specificity is quite specific between the same range of species. Longevity means that you can go with long time, but as Professor Mazzaroni mentioned in the earlier discussion, that we need to understand also, we don't know yet how long the DNA of self-DNA can be accepted by the plants or accepted by the organism. And another quick reminder is that a part of self-DNA, the DNA that inhibits the growth of its own organism, we have also the concept of no-self DNA, meaning that when you go around in the soil, you have a decaying plant, and this decaying plant releasing its own self-DNA, but you have also other organisms around that also decayed, and then they release also their DNA to the soil of the environment. And that DNA, the red one, the one that is not originated from the decaying plant, will also have an influence to the plants nearby. So that is what we would like to understand between, when you talk about organism, you talk about homeostasis, the balance between the good and the evil, the balance between the self-DNA or no self-DNA. But I did not say that the self-DNA is bad, no. Because what we know right now is that the mechanism is inhibitory. But maybe we can find, maybe in the next future, another mechanism that perhaps even more complicated than this one. I think Professor Mazzaroni already talked about this also, I'm quite sure. So we need to understand that in the world of science, what we know now may be changed in a few hours or even days or months or years later. But the concept of this self and non-self DNA, meaning that if you try to understand the no-self DNA concept, is an inhibitory mechanism of a DNA that comes from the non-conspecific species into the targeted path. I will explain to you in the next slide. I think I forgot to ask Dr. Romney, right? You asked about how the nucleic acid can be sent by the organism. So you talk, this is from the Nature Review by Slee and Hartman in 2016, talking about not only RNA sensing, this one, double-strand RNA or single-strand RNA, but also double-strand DNA sensing, meaning that all mechanisms are possible. What you need to understand is that this is the case in the human or even in animal. When you have double-strand DNA, depending on the receptors, even though the receptors are normally related to the immunity system of the organism, when you have this double-strand DNA, it can trigger many mechanisms. For example, immune sensing receptors or even nucleic acid receptors. And then you have a lot of aspects that is going on the organism. When you talk about this in animal, I think the same in the plant. Maybe the mechanism is not quite, maybe 100% alike, but the concept should be the same. Because you have receptors in every organism, and then you have activities. So that is why in our laboratory, we try to understand this concept. So this is thanks to, for that moment, we have a research group, what we call Alibi, aligning bioinformatics, and also working closely with PT Ekotalis Technology Indonesia, and also by Pharma, BV, and corporate system. We try to understand the mechanism. We try to duplicate or try to reprove what Professor Mazzarini's and team has been done in Naples. So what we basically do, we work with two species. The first one is Oryza sativa, rice. Second one is the oil palm. The next slide I will explain to you about the oil palm. But first, with this rice seed, we try to treat them with three applications. First one, we only applied with this cotton under small packages using a distilled water. It's sterile water. And then we try to wait for the growth for 5 days. But actually, we continued the counting until several weeks until the plant died. But in 5 days, you already can see that there is some changes. The second one, we use Noself DNA using the Xanthomonas DNA. Xanthomonas is the pathogen that attacked rice. So it came from different aspects of microorganism, and then the last one is self-DNA from its own rice leaf. What you can observe is that there is a normal growth in the control. No problem, everything looks well. But when you see no self-DNA using Xanthomonas DNA, there's something changed. You see, this one, the green one, is what we call plumula. Plumula is the leaf. And the white one is radical. Radical is the root. What you can see here is that you have particularly small numbers of plumula, but very long radicals. But when you apply itself DNA here, you see there is a very low and slow growth of the even radical or also the plumula on its own self-DNA, meaning that the theory, the previous 3 slides, has been proven here. So we believe that something happened here in no self-DNA using Xanthomonas that triggers something in the rice seed. So the rice seed order the cells to grow and to maybe try to seek nutrient for defending themselves. For example, this is a theory. That's why the roots grow very long. And the next aspect is we learn in the Elais guinensis in oil palm. We use a seed from the oil palm. We experimented the same thing using no self-DNA, this one, self-DNA, using its own famous pathogen, Ganoderma boninensis, and using also the environmental DNA. What we call environmental DNA is that it's a combination of a lot of DNAs in the soil. So we name it soil DNA, and then we have this normal growth on distilled water, and then this one self-DNA using leaf from the oil palm. So we use both leaf, so that the reason is that we need to compare between rice and also between oil palm. What you can observe is that you observe the same tendency or mechanism. When you have no self-DNA, you have enhanced root growth. When you have environmental DNA, you also have enhanced root growth compared to the normal growth in the distilled water. But when you apply self-DNA using leaf from its own species, you have approximately undergrowth, differences in few centimeters of long and numbers of lateral roots. So meaning that the no self-DNA gave again a different phenotype compared to the self-DNA. So using these 2 small and in vitro experiments, we would like to prove that the no self-DNA mechanism could be beneficial to the organism that we apply to. And we would like to come back, make sure if really true that the self-DNA itself can inhibit its own organism. So we use both of the pathogens, Ganoderma, this one, Ganoderma, and also Xanthomonas. We use a small microplates with hundreds of tubes. So we take pictures, only 6 of the tubes, it's very small. We use spectrophotometry technology to really make sure that the obscurity of the liquid is not based on the media, but based on the growth of the pathogens, of the microorganisms. So what we can see here, this one is controlled. When you see yellow, because there is a peptide in the media, so you see yellow, but actually it's sterile. And then we apply sDNA here by its own. This one, so sDNA from Xanthomonas. This one, sDNA from Ganoderma. And this one, we did not apply sDNA, we just put distilled water. Actually, the same with the control, but the control is only media. So what you can see here is that visually, you can see changes that, especially in Ganoderma. This one is mycelium, right above the liquid. When you have a liquid media, you have fungi, it tends to grow in the upper level of the media. But when you see here in the sDNA of the Ganoderma, the growth was inhibited. The same happens to Xanthomonas, the growth inhibited. It can show by the curve that we observe in our laboratory that has a decrease of number of colony. But when you plate them in the petri dish, the bigger petri dish, you can see that when you apply sDNA, this one you cannot see, but you have full colony. Every inches of the petri dish full of Xanthomonas. But when you apply sDNA, they tend to grow really slow, meaning that the sDNA concept is inhibited the growth of corn-specific microorganism, meaning that its own DNA inhibits the growth of itself. And how can we direct the Noself DNA technique? So many scientists, I say many because there are groups of scientists, a part of Professor Mazzoleni is also trying to understand this concept, maybe a bit different understanding, but the way they treat the experiment looks alike. For example, Feriskia Jimenez from Mexico, tried to put Phytophthora capsicum from pepper. From pepper, try to extract the DNA and try to fragment it, even though they use a different method from sonification, and they drenched the application of this Phytophthora capsicum to infect the pepper. And they saw a lower disease severity and reduction of plant immunity, meaning that this theory should be improved and should be able in the future to be applied in the bigger level. And the question is that, because when you talk in vitro, you talk about what we call aseptic environment. You only several aspects that you can control. But when you go to the environment, not environment, just go to the greenhouse, like this, you will see a lot of things like mixed organic sources, crop wastes, that you need to understand all of that. You can extract the DNA and you can call it environmental DNA because it comes from different species and we don't know the effect of this environmental DNA. But Carvajal and Valenzuela tried to understand this and give the opportunity to create an experiment from it, try to induce the plant, what we call plant immunity. So the concept is basically like when we have a vaccine of COVID-19, so that in the future we cannot get the disease to be severe. So the question is that us, as a scientist, we need to pay attention to the dose of the environmental DNA. I think that is the question of the day from fellow scientists. How much enough and how much is too much? That we need to understand importantly. And from the study of Professor Mazzoleni's and team and other scientists, we understand also that feasibly extraction protocols need to be established. We understand that when you have application from DNA, you need to extract the DNA. How much you need to extract? If you want to apply, for example, in our institution in Oil Palm Research Institute, you have hectares of plantation. You want to apply that, how many DNA you will need, and how to administer that. That is the one that we need to think in the larger scale. The second one is that the utilization of this Noself DNA can be applied to handle plant pathogens. The third one is that how to see that it's an extremely important concentration dependent. 200 ppm, 300 ppm for this species and 400 for that species. And that is the one that we need to think about. So Marcelo Janus said to us earlier this morning the importance of factors, maybe microalgae, why not other microorganisms, why not other particles, for example nanoparticles. If you have expertise on that, you can try to experiment on that and maybe we can see a good result. And massive duplication of DNA, maybe we need NGS machine, but then when you talk about business NGS machine, it becomes quite expensive because you need to produce something from expensive machines. Then when you goes on commercialization, should think about how the BEP will come back to us. And maybe to end the discussion, I would like to maybe try to give you a perspective of how we use this technology in the future. Maybe I will talk first in the blue line. So the blue line is quite clear. So the theory has been proven. We need to also enhance the experiment on a larger level. But we know that maybe we can use this Noself DNA application to try to overcome the problem of Ganoderma in oil palm or even Xanthomonas in rice that has been devastating for the last few years by developing Noself-based product. And the other one is how we think about the management of the oil palm plantation, for example, because we tend to use chipping and sunning for replanting. And I talked to several experts this morning and they don't know how long we should put that biomass there, 2 weeks, 3 weeks, 5 months. Some of the plantations put there for 2 years or 3 years without doing anything. So we put back a lot of biomass and of course we put back a lot of self-DNA in the soil. So I think that is all from me, and I would like to quote Albert Einstein, reminding myself as a scientist that creating a new theory is not like destroying an old barn, but actually it's like a mountain gaining new and wider view. So I think that maybe Noself DNA is still new to us, but I think we should open our mind and I don't know, the research is always something new. Okay, thank you very much.
Thank you very much, Dr. Risa Riputranto, who has delivered the topic regarding the proof of concept about the application of Noself DNA to mitigate or combat the severe disease on both major crops in Indonesia, such as oil palm and rice. And he has shown also the potential use for Ganoderma in oil palm and Xanthomonas in rice. However, he also shows something that we got to take care about, because his finding will turn around the current practice, especially for the use of compost that originated from the same material of the biomass. This is something that's very challenging. Thank you, Dr. Isa. And for the second speaker of this afternoon, we do have Dr. Romi Roviadi, who is going to share that something different with Dr. Isa. It is related to fishery in Indonesia. You got 20 minutes, Dr. Romi.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. First of all, I would like to say thank you to Ecostalis Indonesia for giving me this opportunity, and the organizing committee for giving me the golden place like this to present my recent findings on the self-DNA and the use of this concept to inhibit the growth of the vibrio in aquaculture production system. Alright, probably some of you do not know what aquaculture work, because we're talking about plant and agriculture before. So I just want to draw you several attention about the aquaculture and fisheries work. We are the maritime axis right now, and we know that we have more than 17,000 islands and 108,000 kilometers square of coastline. We have more than 275 million people, and the majority live in the coastline. And we have 12.1 million tons of the fisheries production. And our ministry said, in order to build and establish the industry, aquaculture and fisheries, we need to make it balanced between the economic and also the environmental situation. So this is the goal for the fisheries and aquaculture production system in the future. So globally speaking about aquaculture, before we go into detail about the self-DNA concept, we know that in the future, 2030, World Bank already stated that 2/3 of the seafood product will come from aquaculture. And even the economists say that aquaculture and not the internet represent the most promising investment opportunity in the 21st century. And look at the data from this FAO. We know that the aquaculture right now is already produce more seafood compared to the capture fishery. So aquaculture is the future. So and this self-DNA probably can be a part of this grow production system. And this is about the Indonesian aquaculture production. Here we have in total, in summary, we have an increasing value, increasing number from 4.3 billion tons. Right now in 2020 is of around 5.2 billion tons. But there is a challenge on how to optimize the production system. We know that with the rise of the fuel right now, aqua feed, the feed has become the first challenge because I already talked with several feed millers. They will increase again the price of the feed in this October. So this is make the life of the farmers become difficult. So the aqua feed, the cost is become the first challenge, and the disease. Disease is the second challenge in order to optimize the aquaculture production system. Market price, because of this volatility and fluctuation in price, also become a challenge. And broodstock and another feed. So quality of the feed is also become a challenge in aquaculture production system. So among the infectious disease, there are a lot of disease: viral disease, parasite disease, bacterial disease. Vibriosis has become a cause of serious losses in aquaculture. This disease reported as a significant challenge to the expansion of the aquaculture and also cause economic losses, more than $100 million global wide, and high number of farm closed, and large number of job losses, and will take years to recover because of this disease, the Vibrio. So this is just to give you the idea about the number of Vibrio during the white phases syndrome in the shrimp. We can see that the number of Vibrio in the normal situation without the disease incidence, this is lower compared to the number of Vibrio during the white phases syndrome. So if we look at the intestine status in the shrimp that culture in the normal situation compared to the shrimp that culture in the white phases disease outbreak, we can see the high number of the Vibrio compared to the shrimp that culture in the normal environment. This is very interesting because Vibrio can be act as the primary causative agent or become the secondary causative agent. And the current practice to manage the Vibrio, there are several practices right now that we can apply, the farmer can apply. So there is a prophylactic approach. We can use the immunostimulation, vaccination, but vaccination not for shrimp because shrimp is only rely on the non-specific immune system. This can be applied in fish, probiotic and corn quenching. Biosecurity is also to minimize the disease incidence, feeding management, and also the water quality management. So there's a lot of water management system, the biofloc, semi-biofloc, autotroph, and all of these things about how to manage the microorganisms in the culture environment. So we're going to self-DNA. We're already talking about the self-DNA, about the functional role of the self-DNA to also to inhibit the growth of the pathogen. So we have a thinking, that idea to assess the species-specific inhibitory effects of the self-DNA from Vibrio harvae and also the Vibrio parahemolyticus, and also to test the species-specific inhibitory effects of the self-DNA to the growth of Vibrio inside the Artemia. You know, Artemia is a very important live food in aquaculture, and other than the like the microalgae, rotifer, Artemia is the most zooplankton that used in the hatchery right now in aquaculture production system. So this is the material and methods that we developed to prove the concept of the self-DNA in the aquaculture. So we have a pure isolate of the Vibrio harvae and Vibrio parahemolyticus thanks to the Gajah Mada University that provide this isolate for us. And we extract the DNA. This is a normal extraction process. And after we extract the DNA, we electrophoresis the DNA, and then we come to the sonication. We cut the length of the DNA from 10,000 Besper to 100 Besper with these tools. We use the ultrasonicator machine, which function to fragment the DNA. So we fragment the DNA into small size from 10,000 Besper until 100 Besper. So this is the agarose gel looks like. So before, when we extract the DNA, when we extract the DNA, we have 10,000 Besper, the length of the DNA, and then when we fracture them with the sonication process, then we can cut the DNA into 100 Besper here. And for the in vitro test, we developed this protocol. We use several dilution. So after we fragment the DNA, we also measure the amount of the DNA with the nanodrop spectrophotometer, and then we got the number of the DNA of the self-DNA is around 96.10 for the Vibrio parahemolyticus and 53.33 nanogram per microliter for the Vibrio harvae. And then we make a stock solution. The stock solution, because we have 100 microliter of the zobel medium, and then we add another 100 microliter of this self-DNA. So the concentration become dilute into half. So the final concentration of the self-DNA is become 48.05 for the Vibrio parahemolyticus and also 26.67 nanogram per microliter for Vibrio harvae. So we put this in the dilution factors, and then we use this well to see the inhibition effect of the self-DNA. So this is the in vitro test using the Vibrio parahemolyticus. You can see, as you can see in the slide, this is the control without the self-DNA. When we put the Vibrio harvae, the Vibrio parahemolyticus inside the zobel medium, they grow fast because this zobel medium is a specific medium for the Vibrio. But when we add the self-DNA, the self-DNA here, we add with the 48.0.5, there is an inhibition. So they suppress the growth of the Vibrio parahemolyticus inside the medium significantly lower compared to the control treatment. And then there is another concentration that because we have 8 dilution factors, then the second dilution can also inhibit the growth of this Vibrio parahemolyticus. But at the end, this is something that I asked to Professor Stefano this morning. What is the minimum amount of the self-DNA that can work well to the inhibition factor? Because our trial to prove this concept, when we go down lower concentration level of this self-DNA, so the inhibition factor is also going down. So the number of the pathogen is keep growing and growing. But with a certain amount of the self-DNA, we can see that there is an inhibition here. So we also prove this concept in the Vibrio harvae. So in the Vibrio harvae, we also have the control without the self-DNA, and then this 26.67 of the nanogram per microliter of the self-DNA, and we go for several dilution also. And here we can also see there is an inhibition here with the Vibrio harvae with this amount of the self-DNA. And then also there is even statistically, this is not significantly different, but in terms of the numeric value, there is a difference here between compared to the control treatment without the self-DNA. So it works. It works, the self-DNA, to inhibit the growth of the pathogen. And this is very interesting for us. Well, and then we also think we do not want to stop until here in vitro test. We want to go further, because aquaculture is really big industry, and we need to take a part to this industry and then prove this concept into the industry. So we are thinking to use the Artemia. As I said earlier, Artemia is a very important live food in aquaculture during the hatchery, during the larval stage, and they are filter feeder. They consume everything, including the bacteria. So it is what it is. Whatever you feed the Artemia, that is come to your larvae. So when you feed the Artemia with the bacteria, then you also feed your larvae with the bacteria. So this is why we use the Artemia also as the important live food and also as the non-selective filter feeder. So before we go to the Artemia, this is just to show you the visual. There's the quantitative number. This is the visual with the Vibrio parahemolyticus and also Vibrio harvey. You see that if the color remains blue, meaning that there is an inhibition growth, but if the color changes from blue into the pink color and colorless color, there is no inhibition. So we can see this actually it can prove the concept that there is an inhibition. This is to confirm the result of the quantitative number here in they remain blue here for Vibrio parahemolyticus, meaning that there is inhibition factor there with the self DNA. And also the Vibrio harvey, these two concentration levels also have the inhibition effect to the growth of the Vibrio harvey. So this is what I'm talking about. So in order to get a very good design of research, we culture the Artemia in the axenic condition. Axenic condition meaning that there is no contamination. Only that type of bacteria that we want to investigate that are present in the environment. So all equipment, including the Artemia cyst, including the culture water, including the equipment, we sterilize them. So there is no contamination there, and we use the axenic larvae Insta2 nuclei to prove the concept of self DNA. And this is the axenic condition. After we harvest the Artemia, this is the Insta2 Artemia looks like. After we harvest them in the sterile condition, and then after we decapsulate the Artemia, we immerse this Artemia in the self DNA, and then we challenge them with the Vibrio parahemolyticus with the initial concentration. So we have 48.05 and Vibrio harvey 26.67 nanogram per microliter. And then we challenge them with this pathogen and see what is the number of this pathogen inside the Artemia. So this is the challenge test. This is very interesting. So this was performed by my master student in Gajah Mada University, and they see that the survival rate of the Artemia immersed in self DNA, they have a better survival rate compared to the Artemia that do not immerse in the self DNA. So also in the Vibrio parahemolyticus also they have a good survival rate, better survival rate compared to the control treatment. And about the density of bacteria during the challenge test. So we also observe, we collect the Artemia in several time points. We see what is the density of the bacteria during the challenge test. We can see here that the density of bacteria in the Artemia without self-DNA is higher compared to the Artemia that immerse in the self-DNA. So this is the density of bacteria during the challenge test. This is with the Vibrio harvey, and this is the Vibrio parahemolyticus. So from 0 to 12 hour until 48 hour, the density of the bacteria is always lower in Artemia that immerse in the self-DNA compared to the Artemia that do not immerse in the self-DNA. So the thing is, we not prove the concept, but we already apply the concept. So we can see the concept is working in this aquaculture production system. And this is, I think, it's become the new approach. New approach how to mitigate this disease in aquaculture. Because, you know, aquaculture is really unique because they stay in the water. There are a lot of pathogens there. So why we not save the farmer by, you know, increase the survival rate and also apply this concept in aquaculture production system. Right now, farmers, especially for shrimp industry, if you ever heard about the disease outbreaks, there are a lot of disease in shrimp industry, and farmers are waiting for the solution. Antibiotics is not the solution. So, but we have to move further in the safest way, how to save the farmer, how to increase the survival rate, and how to make a better, good return on investment in shrimp industry by using this concept. So, future study. Because this is also interesting. If this is work in the invertebrates, because Artemia is invertebrates. Invertebrates means they do not have the non-specific immunity, meaning that they cannot produce the memory. They are very primitive animal. But this concept can be worked in invertebrates. How about fish? Fish is vertebrate. They have a specific immune system. They can produce the memory. Probably the result will be different or probably better. So this is very interesting because our aquaculture, as I mentioned earlier, aquaculture in the future will produce more seafood. We are relying on aquaculture to get, because this lunch we already get fish, a lot of fish, and in the future it's always coming from aquaculture. It's not from capture fisheries. But this is should be start with a good milestone, a good step that we provide a solution to the disease, because disease can cause highest number of mortality in aquaculture. So I just want to highlight about the Vibrio because they are the opportunistic bacteria. We also analyze about the communication thing about the bacteria. This is what they call as the quorum sensing. And there are several active substances that can also inhibit the communication of the bacteria of the Vibrio, what we call as the quorum quenching. So probably this is more advanced because we not only inhibit the communication, but we also inhibit the growth of the Vibrio in the aquaculture production system. So I think that's all for my presentation today. I just want to thank to Gajah Mada University as our collaborators and Ecotalis Indonesia for collaboration. Thank you and thank you.
Thank you, Dr. Romi Nofriadi, who has shared to us the current update of finding on the Noself DNA potential application on the fisheries, especially for shrimp with related to the Vibrio disease. Our last speaker this afternoon session is this kind of a reflection of what we have heard from this morning until this afternoon. Will be presented by Dr. Joko Pamungkas with the title of Reflection on the One Health Concept for Animal and Human Diseases. Dr. Joko, the time, 20 minutes.
Thank you, Professor Didik. Good afternoon, everyone. Assalamualaikum warahmatullahi wabarakatuh. The title given to me is about reflection of the One Health in considering the application or implementation of the Noself technology in the One Health concept. And probably, as the name implies, I will be the last speaker's Pamungkas. And I probably have to start with the outline, but I will go very briefly with the definition of the One Health. I don't want to give an impression that I teach a basic knowledge of One Health. I believe that you all are aware about this concept or approach. And then also in relation with the zoonotic diseases, wildlife is very important in the One Health systems. So basically, we also would like to point out about the importance of wildlife. Also, that in some aspect of the health, both for human health, animal health, and ecosystem health, we also see the benefits of applying or implementing One Health approach. And then I probably will give some success story of the global examples of One Health approaches that's been implemented also in Indonesia. And last but not the least, this is the very important things and also new in this approach or technology of Noself mechanisms. So basically, we also try to formulate some questions for the future application or implementation of Noself technology in One Health concept. Okay. For the definition of the One Health, we try to have the newest version of this definition. Actually, it's been developed by tripartite in the past, consisting of the WHO, FAO, and also the OIE. Now is called the World Organization for Animal Health. So the tripartite actually now joined by another organization, the UNEP, UN Environment Program. So then they actually have the quadripartite program. As a visual definition of the One Health, basically on the left circles, there are the key sectors and also disciplines that are taking part in the One Health concept, such as the human health or human disciplines, and also the animals as well as the environment. So the sectors, the three sectors are actually taking the most critical part, but we are not ignoring, we are not eliminating the other ministries or the other sectors part other than these three key sectors. And then also, we also need to emphasize that this concept is not going to have benefit if it is only in the national concept or in the national level, but we have to also apply or implement in the subnational and particularly in a very, very spot or location that might be impacted by the interactions of the human, animal, and environment. And then the circles in the middle is the outcomes of this approach, that we would like to achieve the healthy humans, healthy animals, also healthy ecosystems. And by having all these outcomes, we need to also consider the 4 C's: collaborations, communication, coordination, as well as capacity building. So basically, we just want to give you ideas that nowadays we cannot just work alone. So medical doctor will not be able to actually solve human or public health issues without the help from the other disciplines or professions. Okay, so Indohun, not Indohan, probably. So basically, Indohun is an abbreviation from Indonesia One Health University Network, and it is part of the Southeast Asia One Health University Network based in Bangkok. Other than Indonesia, there are 8 now OGUNs in Malaysia, Thailand, Philippines, Myanmar, Vietnam, Cambodia, Laos.
Recording in progress.
We have about 20 universities in Indonesia as members of the INDOHUN, and out of these 20 universities, we have about 30-something faculties or schools. And out of the 20 universities, some already form or establish the One Health collaborating centers, like the University Gajah Mada University also has the One Health collaborating centers, and secondly, Udayana University in Denpasar, and third, the Erlangga University in Surabaya. The fourth is Kuala University in Banda Aceh, and the last one is the Cendrawasih University from Papua. And probably this year we will have the 6 OHCC, One Health Collaborating Centers, from IPB University, and next year probably from the Hasanuddin University, South Makassar, South Sulawesi. Okay, so One Health is not just about infectious disease. Again, it is much broader than infectious disease. It's also inclusive the communicable disease, non-communicable disease. But I just want to point out here that talking about zoonotic diseases, so we have to consider about several facts that there are some infectious diseases that only affect the human beings, and there are some only affects the animal beings. But also we have some zoonotic diseases that will affect humans as well as animals. It can be transmitted from animal to humans as defined by anthropozoonosis, but it can also be transmitted from human to animals defined by zooanthroponosis. So in this case, we need to understand, we need to learn more about who is playing as natural host or reservoir and who is playing as fetal host. In many cases, the human beings are the fetal host from the reservoir role by the animals and also especially from wildlife. This again, another terms that probably is also heard very often, emerging infectious disease. So 75% of the emerging infectious disease in human are originated from animals. So this is the one that we call zoonotic diseases. And then out of these zoonotic diseases, about more than 60% associated with wildlife. This makes important that the approach must be a One Health concept, One Health approach. This is a definition by the National Institute of Allergy and Infectious Disease, US NIH. Infectious disease that have newly appeared in population defined as an newly emerging infectious disease, and then the infectious disease that have existed but are rapidly increasing in incidence or geographic range within certain period of time. This would be the re-emerging infectious diseases. These are some points that makes us realize why wildlife are important in the One Health concept or One Health approaches. So again, more than 70% of emerging zoonotic infectious disease have wildlife origin. So if we try to give some examples, in 1981, we understand that the first case of AIDS are identified in human beings in Los Angeles. And then 2 years after that, 1983, the virus was identified, HIV. And then later on, maybe about 3 or 4 years later, SIV was isolated. SIV is the simian version of the HIV, the retrovirus. Simian immunodeficiency virus was isolated later from non-human primates. But when we try to actually look further in the bioinformatics systems, the scientists actually proved that the SIV existed earlier than HIV. And SIV originated from non-human primates, especially from African species like chimps and also the talapoid monkeys. And more pathogens actually are spilled from the wildlife to livestock, from the wildlife to human beings, or maybe from livestock to human beings. And this is because of the human activities creating more opportunity for the changes of types of contact that probably the several industry, mining industry that has to open lands, and also some development that has to actually open the forest. So basically, there are some loss of habitats of this wildlife. So the wildlife try to find new habitats, and basically there will be some interaction between the wildlife and the livestock as well as with the human beings. And so far, probably predicted there are more than 1 million unknown viruses in this earth, on this earth, with some pandemic potential that might occur in the future. And only 1% of this more than 1 million viruses has been identified so far. So wildlife actually are very valuable sentinels for threats for human as well as for livestock, domestic animals, including the non-communicable disease threats. And there are certain evidence or proof that this wildlife is quite important for the possibility of disease contracting to human beings. This slide shows the importance of the three taxa, the three orders of animals. Compiled by Kevin Noself from EcoHealth Alliance. Basically, he compiled publication from several articles, maybe several decades, maybe 4 or 3 decades, and then he concluded that despite of the total viral richness in every orders of mammals, the three taxa are playing important roles in contributing to disease transmission to human beings. Those are the Chiroptera, the bats, yeah, and then the primates, and then the rodents. Yeah. So these three orders are the most important wildlife to be considered as a carrier or as a reservoir for some diseases for human and livestock. So we are not supposed to blame these animals, yeah, but we have to be very to have a wisdom, yeah, that knowing that they are harboring some infectious disease pathogens doesn't mean that we have to destroy them, but we have to actually make our environment to live harmonious with these animals. This model developed by Dr. Billy Carres from 2012, yeah, published it in the Lancet, and this is a normal situation that probably should be paid attention by us, every one of us, yeah, that wildlife are shedding viruses in the environment. And that will be some fluctuation of the shedding. And at some time point, it's possible that the shedding of the virus might spill over to human beings, yeah, or to livestock. Yeah. And this will cause some economic loss and probably fatalities and mortalities in the human beings or the animals. So basically, this is a model that if we just take everything as is without any prevention, without any mitigation, so this will occur all the time. We will see that the occurrence of disease in human beings or in livestock, but then after a while we try to study further that this disease actually can be prevented when we understand that the potential viruses shed by the wildlife are identified from the beginning. So basically the second curve showing that if we do some say active surveillance, yeah, so we would like to see how many viruses are there in the environment shed by the wildlife. And we could identify at some time points that certain virus might be harmful to human beings in probably 1 decade or 2 decades from now. So we portray the situation now to predict the possibility of potential pandemic in the future. So if we do that, if we do the surveillance of the virus from the wildlife, then we should be able to prevent the occurrence of the spillover to livestock or to human beings. So the loss would be decreased by 60%. This is a model, of course, yeah. But in the reality, there are lots of factors that we might have to face. This is a good example. Yeah. World Bank actually already made some value evaluation of the disease effect, about the economic loss of disease emergence from time to time. Yeah? And we see here that lots of loss that's been reported, and the last one from COVID-19 is very huge, about 15 trillion US dollars. Yeah? So this is the loss that we face if we just ignore if we just take the activities and business as usual without any prevention, without any control nowadays. So this one model again, this project is called PREDICT, and it was funded by the USAID, US Agency for International Development, with the consortium from the US consisting of the UC Davis, EcoHealth Alliance, Metabiota, Smithsonian Institution, WCS. And it consists of more than 30 countries involved with this surveillance, including Indonesia. So we basically took part in the first stage of this project from 2009 to 2014, and then continued from 2014 to 2019. From the one decade project, PREDICT globally identified about 1,100 viruses, and more than 900 are novel. And it consists from the 28 families of virus, and this involved the samples from wildlife, from livestock, as well as from human beings. Not just the virus discovery, but PREDICT project also have some capacity building. So basically, lots of more than 6,000 people actually been becoming like a workforce for this project, and then more than 60 laboratories in the world has been built the capacity to be able to detect the virus that previously undetected, and then more than 160,000 also operating, and more than 1,100 viruses identified. A bit about the project in Indonesia, we also try to have some behavioral studies in human beings, and as we probably are aware, some part of Indonesia, and again this is not just specific to North Sulawesi, but also happens in Kalimantan, in Sumatra, and somewhere else, even in Java, like Yogyakarta, Solo. Yeah, a lot of people consume the wildlife meats, the bush meat. Yeah, so we can see that the three taxa of the important wildlife that contributes disease pathogen to human being actually are consumed very regularly, especially in the northern part of Sulawesi, like the bats. These are the roasted bat, not really well cooked, yeah, not yet well cooked. But we also sometimes see primates, non-human primates. The 7 species of non-human primates in Sulawesi are actually critically endangered, and they sell this non-human primates carcass for very low price, like 50,000 rupiahs per kilo, and it will probably sell for maybe 250,000 or 300,000 maximum. So compared with the conservation status of this animal, it's very ironic to see this situation. And it's very common that they actually have this sold in a wet market in North Sulawesi. Again, I don't want to what do you call it? Discredit the situation only in North Sulawesi, because we just focus our activities in North Sulawesi, but it also happens probably somewhere else in Indonesia and also Southeast Asian countries. Okay, so some recommendation that we also give that we have to have a massively scaled up One Health surveillance across the 3 interface, the wildlife, livestock, and human beings. Yeah? So lessons learned from the PREDICT project, we should also try to have disease surveillance in more stable situation. Basically, it is important that the country ownership of this surveillance is a key. It's not going to be sustainable if we always depend upon the international agencies' funding. So basically, we have to ask attention from our government to provide support for continuous surveillance. And of course, with the variety, diversity of biodiversity and also cultures in Indonesia, the risk is not uniform from one land to another land or from one island to another island. So basically, we should have some kind of target surveillance to be implemented from one place to other place. We have to identify and to strengthen our One Health workforce from field to laboratory. At the moment, this PREDICT project actually try to strengthen the capacity of the laboratory from the government of Indonesia. So 8 national laboratory for animal health or animal disease investigation has been trained for this particular laboratory protocols, as well as maybe 4 or 5 units from Ministry of Health, and several other also laboratories from the universities. These are the field activities that we conducted. So we take samples and we try to preserve. We don't want the samples to be ruined from the transportation or from the poor storage in the field. So we bring all the logistics, required logistics to field. It is not easy. Sometimes we have to also have to go on the water, yeah, not just on the road. This is almost the last slide, so I just want to point out here that common viral transmission from wildlife to human beings or to livestock are taking a certain patterns, and we would like to show you here that this reservoirs basically are animals that are infected by certain pathogens, but they are not showing any clinical symptoms. They're not affected by the microorganisms or the pathogens, but once the same pathogens actually infect other wildlife or maybe some livestock, it will give some clinical symptoms and probably some fatalities as well. So these are the intermediate hosts that also play a role as an amplification or amplifying host. And human beings or livestock maybe is the fatal host for all these situations, that it might be transmitted directly from the wildlife to human beings, but at many cases it also has to go through the intermediate host and then infect the human beings. And from this slide, we actually have several questions that we learned today about Noself technology. The first question is how the Noself technology could be implemented in preventing emerging infectious diseases. I think I learned from the discussion earlier that might be we have still to think about viral RNA that might be involved in many cases, that most of the emerging infectious disease actually caused by the RNA viruses. And then we also need to consider maybe this will be able to be implemented in non-communicable disease, as well as on the antimicrobial resistance that's been discussed also earlier today. And then we probably need to know which chain should we pay attention to, either to the reservoir or intermediate host or the fatal host. And this, of course, will need some approaches that, again, in the importance of the sake of importance for the human host, probably we will focus on the fatal host, like human beings or livestock. And then we also have come with the last question on the stage of disease control and prevention. Will this be comparable to something immunization that probably been introduced in livestock or in human beings, or it might be something different, but will what do you call it? Complement, to the current situation. So again, I'm new in this technology, in this mechanism, so maybe the question is not quite representing the real situation for every Indonesian, for every person here in the room, but this is something that we can discuss further. With this, I will end my presentation. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Dr. Joko Pamungkas. Ladies and gentlemen, we already have some additional complete overview regarding the problem of health in the global stage. And from what Dr. Pamungkas has presented, we could learn something that how complicated this, what they call it, the human disease originated from animal or zoonosis. But we also noticed that he should put control on operation and also control on benefit to people to minimize the negative impact of this kind of disease. But on the last presentation, he raised challenges on how the application of Noself technology to overcome this kind of disease who currently also we face in the global state. I think this end of the presentation for the whole day seminar for today. But for the three last speakers, I would like to give some opportunity for the audience to raise some question or comment before we move forward to general discussion that will be led by Taku. Any question or comment, please? Okay, please, on the back, gentlemen. Maybe you need wait moment with your mic. Please identify yourself and to whom the question is.
Thank you. My name is Haryono Joyediharjo. My background is actually aerospace engineering, but I come here because of friends, and I got to be attracted. Then I'm attracted to the topic that is very, very what is intriguing. First is One Health, and the second is Noself mechanisms. Both are actually very new, but I learned actually that One Health is already, it's a universal, promoted, although it originated probably in the last decade, probably, something like that. But what I'm really very curious is about the origin or the philosophy or the basis of the technology of what you call Noself mechanism. I'm sorry I came late, so I didn't really follow everything. But of course, I'm still learning, so I would greatly appreciate if that can be summarized, probably also for the benefits of other attendants here. Thank you very much.
Thank you. Pak Joko?
Haryono.
Okay. Anyone would respond this question? Oh, one by one, go ahead.
Thank you, Pak Haryono. I try to give my best answer. But we have Professor Stefano here that can provide a better answer than me, and also another speaker. But for me, to best to my knowledge that the concept, Pak Aryono, the Noself DNA, is based on the recognition of these foreign materials, the DNA, but they can act in the species specific to inhibit the growth of that type of species of bacteria or DNA. So the thing is, they work into specifically to the DNA that we fragmented. So that's the concept of the cell mechanism. So this is a novel approach in how to mitigating the disease rather than to use the antibiotics that can have the antimicrobial resistance in the tissue of the culture organism and other or plant or human. And by doing this I think is more safe to the human because this is no reaction to the organic to the organism, and this can be applied in the future as the novel approach for One Health solution. Thank you.
Dr. Isa, you have something to add, please?
May I proceed? It's very interesting because I remember there's a professor in, I think, ITB who develops how you don't use fertilizer, but use something that originates from that plant itself to have a better growth. Like this is for agriculture, and one of my students have practiced that. Now, in that case, I like to ask further because maybe I do not completely got all the information. But that means that are you resorting actually to the DNA itself rather than to the environment or others in order to take the best advantage of that? In fact, for many positive purposes. Is that so? That you resort more on what is, so it is no self-mechanism, but is it self-mechanism? So if not, what are the differences? Thank you. I hope you got my questions, right? Thank you.
Okay, great.
Okay, thank you, Professor Pak Haryono. I will try my best also to answer as a scientist. So the basis of self and non-self DNA. So basically what you're talking about or asking about is about sensing mechanism. I will give you an analogy. So if you have an organism, so the organism will cope their life from diseases, from, I don't know, stimulus by acting in two ways. The first one is by receiving warning from themselves. That is what we call self-DNA. And receiving warning from other. Because we're talking about DNA, so we talk about self-DNA and non-self-DNA. So basically, when you have either self-DNA or non-self-DNA, in my limit of my knowledge, I do not quite understand completely how the mechanism happens, but either the both mechanism, it can triggers what we call internal system warning in the organisms in cellular level, meaning that in self-DNA, the self-DNA would like to say to your cell, there is something happen, there is your DNA fragmented or destructed or the one that is not normal according to the cell, and the cell will respond, let's stop first. Like when you have an accident in the road, you need to stop and breathe first, right? So that you will understand how to take action after. That is self-DNA. The no self-DNA, meaning that when you have a response, you see other people having an accident and you say to yourself, I need to be very careful to not having the same accident as that guy in front of the road. That is what we call the no self-DNA. But the concept itself can be used also, that no self DNA, meaning that no self DNA for you, but could be self DNA for the diseases inside you. For example, you have plants, oil palm have Ganoderma inside, so when you put the DNA of a Ganoderma, we have potentially inhibited the growth of Ganoderma inside the oil palm. This is only the analogy. So meaning that either way you choose self-DNA or no self-DNA to understand, you are facing with a sensing mechanism that has been given maybe probably a long time ago, and we have the opportunity to understand the mechanism nowadays, that you have this mechanism, you can use it to developing product, for example, if you talk about plantation or even health, developing drugs maybe that can, that based on the self or non-self DNA to induce or inhibit some mechanism in your body, or even the mechanism of other diseases generated by viruses or bacteria and etc. So I hope that I give you a more global understanding using an analogy, meaning that we have a long way to run actually with self and no self DNA. But this no self DNA technology is actually the one that we can think about, because it will inhibit the other diseases that we don't want to in our body, or we don't want to in other plant species, for example, if you, or even in fisheries. So yes, I'm quite excited with what we see today and nowadays with the no-self DNA. Maybe in the future we will find another understanding about this technology. I hope that's all from me. Thank you. Thank you, Pak Aryono.
Thank you, Dr. Risha, and I hope that's already satisfied Professor Haryono for his question. Otherwise, after the meeting, you will discuss again with Dr. Risha outside. Ma Joko, you may have something to add.
Thank you, Prof. Haryono, for the question. And I think I'm going to answer about the One Health concept or approaches. So basically, if we define, if we learn about the definition of the One Health, and then a person probably will think, oh, I've been doing that for so long. I've been doing that from 3 decades ago. Yeah. So individually, the practice, the activities probably been in place, but then because of the appearance or the occurrence of H5N1 avian influenza in early 2000s, more realization, more understanding about the importance of this One Health concept or approach among the institutions. So it is not just being practiced by individual, but it should be also practiced by the organizational level and also even sector from the government. So basically, the concept is something that in order to be able to combat the human disease or livestock disease, it cannot be just practiced by one sector, like Ministry of Health or Ministry of Agriculture, but we have to collaborate, we have to coordinate, communicate each other in order to be able to actually solve the problem comprehensively, not just in the human aspect, but also from the upstream. Maybe we have to deal with the ecosystem, maybe something wrong with the management of the ecosystem, something that we have to think as a holistic situation, but not individually, but more institutional organization, or maybe even government. Yeah. I think that's my explanation, Pak Haryono.
All right, thank you, Pak Joko Pamungkas. And I think we are already 30 minutes over the scheduled time, but there's no problem because this is very, very important issue that we would like to have a response from all audience here who come over to this meeting. What the next of this discussion? Yeah, we will have some discussion for general issue later, that by Dr. Tako Botama, and the target for this session is to make a networking, because Ecotalis Technology Indonesia could not work alone. We need all stakeholders who may have the same interest on the application for so-called Noself technology, for example, for agriculture and also for pharmacies, and for the scope of the global approach is for the achieving a better life for the people. And I think we have come to the last session of this afternoon. Before the general discussion, I would like to ask you all to give a big applause for all our three standing speakers this afternoon. Thank you very much, and I'll give back the time to Ms. Anna Junadi for the session, the next session. Thank you.
Thank you very much, Professor Didik, Dr. Joko Pamungkas, Dr. Riza, Dr. Romi. But before we go to the next session, I guess the weather is getting a bit cold, so maybe we can all bring our cup of coffee in here. Please do. Yeah, so most welcome.
Good afternoon, everyone. And I hope everyone is still awake, because the low temperature has one function: you wake up. But then later on, you fall asleep because it's too cold. Pa Rizaya, please come and help me. Pa Romi. So, here you see in front of you, that's the team from Indonesia and PT Ecotalis, but I'm from Holland, by the way. And very happy to see you all here. You've had quite a menu today on the no-self principle and the One Health. I think I want to thank Didik especially for pushing through initially the idea of combining the One Health concept with the no-self mechanism as found by Stefano Mazzoleni and his team. That was quite a visionary move. But on the other hand, I have learned in my career that the more complicated you make things, the more difficult action becomes. Because we heard Pa Joko, thank you Pa Joko for your presentation, it's very interesting, I learned something new, talking about holistic. And every time when I hear the word holistic, I think, oh, this means nothing will happen. Because holistic means everything at the same time. And I believe that Pa Riza and Pa Romi have shown to us that we don't need to be all the time holistic, although that is a very good idea. We can also start where the business is, namely in oil palm production, or palm oil production, I should say, and in the production of prawns. That is what you guys have clearly shown to us. And to be frank with you, I was expecting many more questions. Now, I have one question. Everyone is talking English, but we are in Indonesia. So, if someone wants to ask a question in Indonesian, or Bahasa Sunda, or Bahasa Jawa, anybody, any language is welcome. Any language is welcome. So don't be constrained by Bahasa Inggris. Yeah. So I'd like to start with a general question, and please consider answering or putting your questions. And this question is to the business community who is represented here. Thank you for coming. So my question to you is, do you have any feeling that what you've been presented today gives you reason to follow up, to further communication? In the direction of new technology, new applications, new management style, Tony, I'm going to ask you a question. We just talked. Can you please give me your impression? Sorry to put you on the spot, but you know, you've been too silent, so you have to speak now. We do it that way.
Okay, thank you very much, Taco. I'm Tony Liwang from Sinarmas Group, dealing with the oil palm. I think this is very impressive technology that we have to start with. Even it's better later rather than never. But I think this is very good point that because now not only we talk about one self mechanism and one health point of view, but today in the global sense, the people talk about circular economy. So not only gaining what the soil can have produced and what the plant can give us, but total circular economy. It means that what we can produce at most from the agro ecosystem. So we are thinking also with the microalgae and all the mechanism that we talk this today. And I think this conference is very useful and very insightful for us. I hope we can start with something real in the field rather than talk only. But don't forget that Indonesia is so wide, so we have to start in some point because and we can start with the fishery, agriculture, even for oil palm and other forestry or whatever. But I hope that from this conference we can start something real in the field as soon as possible. Thank you very much.
Thank you very much, Tony. That is music to my ears. We'll follow up. We'll follow up together. Rajiv Kumar, can I ask you to give your comments? I'd be really grateful, sir. We only know each other today, but very happy that you would come here.
It's our pleasure. I'm Rajiv. I represent CP Prima. So we are aqua company. So what I understood from Prof that most of the research, what have been done so far is on agriculture or on animals, not in aqua field. But being a scientist and you know in front of all the scientists, we are very hopeful and we think it's a new field, concept is good. And Paromi has also presented, you know, on Vibrio parahemolyticus, there are some promising results. So I hope we will have, you know, good work and in the future, the coming, because when we talk about bacteria, if we just take what Romy has selected, Vibrio. Vibrio is the one which is killing the shrimp hatcheries. And now for last 10 years in the ponds also, if you would have heard EMS, early mortality syndrome, or AHPND, which is caused by toxins present in Vibrio parahemolyticus. So yeah, there is a challenge and we have a concept. I think we can go ahead. Yeah, that's from me.
Thank you very much.
Thank you.
May I ask Pariza and Paromi, can you each respond to what Tony said on oil palm and you, Paromi, and to what Parajip just said? Yeah. Can you give us a comment? Here is the microphone.
Thank you, Pataku. Thank you, Parajip, for your comments and your belief to move this forward into the aquaculture industry. Yes, as highlighted by Dr. Rajiv, that Vibrio is we can consider as the primary disease in aquaculture production system, because Vibrio is everywhere. As long as with the high salinity water, the Vibrio is there. Brackish water for shrimp and marine fish with the high saline water, the Vibrio is there. So how to control? We have to make a peace with the Vibrio. I believe that if we try to eliminate them from our cultural environment, I think it's useless, because the replication number and the replication process is very fast. So when we try to
make the environment free from Vibrio, if we still culture the shrimp and also the marine fish in the saline environment, it will be impossible. So the thing is, in the future, I think we need to make a peace with this pathogen. How to make a peace? Inhibit the growth of this pathogen. So I think this is a very good concept in the future for aquaculture. There is also another, as I mentioned in the presentation, there are several techniques to mitigate the vibriosis problem. We can use the vaccination for fish, but not for shrimp. We can use the probiotic, how to make a good competition between the good bacteria against the Vibrio as the pathogen, and we can also use the immunostimulation using beta-glucan. Our research recently said that beta-glucan can also enhance the immune function, but this is requiring a process that sometimes also have a disadvantage to the aquatic organism. But this is go to the organism itself. We go straight to the pathogen. We go straight to the pathogen and inhibit the growth of the pathogen. So I think this is a very good concept in the future for aquaculture. So thank you, Pak Rajiv, for your belief on this. And if there is another people from aqua sector that coming to this conference right now, I think you also agree that vibriosis is one of our main challenges in aquaculture, and we have to deal with it. And I think the SAF DNA can become one of the mitigation tools. Thank you, Pak Tako.
Thank you, Pak Romi. Thank you very much.
Thank you, Pak Tony Liwang. So from what you have already mentioned, you said about the point about circular economy. You said also about we have to start at some point, and I totally agree with you. Basically, when we have in our industry, in oil palm plantation, we have a lot going on. We have a lot also have not been done. We have so many homeworks actually, even though that the industry of oil palm is moving forward. Like in Indonesia, maybe it's one of the most important plant species that we ever have. So to answer your statement, I think I should start with a quote that changes is the only thing constant, that we change every time. So our environment changes also, our plant species change also. Meaning that when you talk about sDNA, it's not only halting in one aspect, developing products, or maybe implementing this product in our oil palm plantation, but also more than that. Meaning that when you talk about sDNA, we haven't talked about epigenetic even. So because you have the connection between what you have in level genetics, and self-DNA is one of that. And self-DNA or even no self-DNA, meaning that you give your environment around the species. If we talk about oil palm, in oil palm plantation, something that we call epigenetic that will push the changes, that will we might see a different responses of the plant. Maybe can enhance the productivity or maybe not, but if we don't try this new terminology, maybe we will never know. So in my point of view aspect as a scientist, maybe first that we need to move forward. I agree with Pak Romeo. So we need to move forward according to what we know today, and starting by researching this aspect maybe one by one, or maybe creating a larger group of scientists that have the ability or have the knowledge or have the what we call the passion in this area of research, and then we can try to create something that even more bigger than this one. In the point of view of industry, we understand that changes in business is something that also common. But the one thing that we need to understand is that we need to prepare what we research today for the future business, right? So what we try to understand in this moment will change how the industry behave in the future. I think that is the point of view of myself, Pak Tony Lewang. Thank you so much for highlighting that we need to start at some point. So thank you very much. Back to you, Taco.
Let's give that a big hand. Well spoken. Then we have guests from Malaysia. Yes, Pak Rahmat, Pak Abdul Rahmat, can you speak? Because I'm very curious to hear what you want to say. Tolong mikro ke dia ya. Very good.
Terima kasih, Dr. Tako. First of all, I would like to thank the organizer, especially you, Dr. Tako, for inviting us from Malaysia. Very interesting subject. This is something new, especially for me personally, regarding Noself DNA. In the interest of food security issue that we are facing now, Malaysia particularly is also embarking into a modern farming. Yeah? As you know, food security involves not only accessible to food or affordability to food supply, but also on food safety. And a country like Malaysia, we are still using a lot of chemical, which is not very safe for the human consumption. We would like to see, or we are very keen to see, the commercialization or the application of this technology so that we could bring it back to Malaysia, or we can apply in Malaysia, because in Malaysia we have two types of farming. One is high hill in Cameron Highland, as you know, the weather is much better, and lowland. In the lowland farming, I'm focusing on cash crop farming first. Two areas or two characteristics. Number one, they are lowland, which has not been proven using chemical-free technology. Number two, they are small plot, they are smallholders. So we are trying to find a solution to these two problems in order to assist the farmers in Malaysia. We have heard about the detail of Noself DNA. I think we would like to get into more detail from Professor Stefano Mussolini and also his colleague moving forward. And this is not the end of it. Perhaps we could also communicate later, directly, perhaps visiting Malaysia, you know, meeting more people and organization, the ministry. I know some of you have met. I think Professor Mussolini have met Minister of Agriculture from my country when he visited Holland a couple of months ago. So that's a good achievement. At least the highest level of authority is already exposed to the new technology. So we are now following up with those progress. At our level, myself personally, we would like to see private sector in Malaysia to take it up. You know, people with money, you know, people who wants to do modern farming. And Malaysia is also trying, just to share with you, we are also trying to be net exporter of food, you know, because we have all the resources. We are very, everything is conducive to be as, at the moment we are not. We are importing a lot of food into Malaysia despite we got plenty of land. I'm not sure about Indonesia, but that's one of the issues, and the government is trying to overcome, trying to do import substitution, especially for cash crop, fruits. Yeah? I don't want to touch on palm oil so much because they are big companies and they have their own organization like MPOB. They are well equipped with money and also resources to do it on their own. But if we can address the problem I mentioned earlier about the cash crop farmers, lowland, smallholders, this is what we are trying to do. So thank you, Dr. Tako, and the organizer for inviting us. We came, a couple of my colleagues from Kuala Lumpur, all the way to attend this seminar. Thank you again.
Thank you, Pak Abdul Rahman, and thank you for your very sincere and open words about Malaysia and the aspirations of the country. I would like to talk about those aspects later on before closing. As you may know, I used to work for the UN, so this is very much, I have seen in the course of my career many communalities between countries, challenges that they actually share. And I completely agree with your word that you would like to see the private sector respond. I believe that to be very, very important remark. And we will see what is going to happen in the future. We hope a lot. We hope a lot. I would like to ask Pak Fasa from Pupuk Indonesia. Maybe you are surprised, Pak Fasa, I'm going to ask you to give some comments. But I'm going to ask you because you represent a very important company and it's public sector. So what are the ideas of Pupuk Indonesia on the topic we have been discussing today? Would you mind sharing your thoughts? Can the microphone please go to Fasa? You have to stand up, Fasa, otherwise they don't know who you are. Sini belakang. Okay.
Okay, thank you, Pak Tako, for the opportunity introducing. My name Fasa from Pupuk Indonesia. Actually, Pupuk Indonesia is a nutrient company. We are focused to fertilizer the soil. So basically, the function of self-DNA is to eradicate the penyakit ya Pak ya, the disease of the plant ya. So basically, if we talk about the company, we have to elaborate with our group because we have Petrosida and Petrokimia Kaya Kutu involved this program. Yes, so this is very interesting because new technology and also new knowledge for us, and very happy to involve this program. And hopefully you also will involve Pupuk Indonesia to elaborate this program. Thank you very much.
Thank you, Pak Fasa. Yeah, that's right. Much appreciated. You know, Pak Abdul Rahman, the phenomena of state companies in Europe under neoliberalist powers, it has been reducing. Here in Asia, we see the opposite trend. It has been increasing. And I will only observe it and not comment. I'm going to ask Jonathan Koppert and Budi, the famous pair from Pekanbaru, to give us their thoughts on how to go about with the algae. You guys, these gentlemen have set up an algae factory, which has been inspected by Professor Mazzoleni, and I would like to hear a little bit of the history, Jonathan, and the way forward, the way you guys look at it. Yeah. This is private sector, by the way.
This is private sector, yeah, correct, Taco. Thank you for the opportunity to share some thoughts. Thank you also for all being here and connecting with you. First of all, I want to make a reference to the slide of Professor Mazzoleni, where he actually talked about restoring a balance in the gut, or restoring a balance in the system which is actually disturbed. And I think that is what it is all about. Also, Professor Romy was talking about it, to restore basically the balance. And that's also what we have been doing with our company Koppert and Biocycle in Indonesia, where we use a lot of biological products instead of chemicals to restore a balance inside the crop. So not to eradicate the problem, but bring it back close to nature. And why I do think this is an elegant solution is also it's very target specific. So it's not like a broad spectrum application. And with that, we also don't do any harm to the diversity which is there. Right now, with a lot of chemical products, they also target actually problems which are not there, or like species of insects which are not there. And I think that's a big problem. And with this solution, we can be really on the spot and targeted. Yeah, getting to your question, Taco, about the algae. So, as corporate, I think we are involved already from an early time on this concept. We've been, let's say, an early adopter or an early supporter. And to get this product basically to market, you also need to be able to produce it on a scale. Because it was also mentioned already, if you want to use a product based on this concept in the oil palm, then you need quite a lot of amount, even if it's only a few grams per hectare, right? Because there are so many hectares. So we started to produce algae in Indonesia in order to be able to provide also these products. As was explained earlier, algae are basically a vehicle, to multiply or to replicate the DNA particles which are needed to have the effect. And that is going with ups and downs, buddy. Sometimes going very well, and sometimes there are some challenges. But we're getting there. And for me, as a guy from abroad, it's of course the cultural difference and the physical distance, which makes it challenging also to make things happen. But luckily, we found very good partners here who can help us, doing what we are doing. And Budi, do you want to say anything? Because I can keep talking, but okay, I keep talking behalf of you. Because I want actually to challenge Taco to find somebody in the audience who can maybe also shed some light, on the application in the field, because we are also bound to certain regulations and certain rules, and to what extent and where does this going to fit on in Indonesia, and what should we think about, because we really want to bring this product to market, and we also want to do it in the right framework. And I'm just curious if anybody can shed some light on that.
Right.
Thank you.
Well, thank you, Jonathan. Those are very clear words indeed, because algae can be targeted. It's specific, it's a product, it's useful in more than one way, we've learned. So is there anyone in the audience who wants to answer Jonathan's question? Is there anyone from the regulatory in the audience? I don't think so. Abdul Rahman, have a shot. You cannot go wrong in answering this question.
Question is very specific to Jonathan Coppert. I know your family is involved in biological solution for farming. Don't you think that this new technology, the Noself DNA, will disrupt Coppert's core business moving forward? Okay, good. Because we are planning to work with you in Malaysia on the integrated pest management, IPM technology, integrated pest management, yeah? Yes, so we thought that, we can do both, IPM technology as well as no-DNA as well. Thank you, Jonathan.
Okay. So we have a long way to go. Thank you very much, Fatul, and thank you, Jonathan, for taking the question. I want to say personally that I have really appreciated and still appreciate working with the big corporate family in all its manifestations, and especially the manifestation here together with Budi, whom I've come to consider like, I'm not sure he's my son, but something like it, something like it. So be careful, Budi. All right. I think we're approaching the end of the day, and I'd like to give Stefano first an opportunity to share with you some of your observations and what you think, and maybe any advices for closing, short closing remarks. And also Marcelo, if you'd like to say something specifically, then the opportunity is now. And then later on, Pak Bungaran, I will ask you to, okay, then I will speak on your behalf. Okay. Yeah, okay, he refuses. Please, can the microphone go to Pa Stefano? There it comes from the back.
Thanks, Taco. Well, first, I just want to repeat that I am grateful for the opportunity and the invitation to come here and to explain my theory and my findings. Not mine, our, because the team now is, the idea started as mine, as a crazy idea. But then it became enlarged to the team, and then now probably we are over 80 researchers in a network connected, doing research on many different model organisms. So I've seen in the last 10 years really an exponential growth in the scientific part. At the same time, the good interaction we had with Coppert helped together with Marcello and M2M to understand better the issue of how to upscale this. Because one thing is, I'm very well aware that one thing is to see in vitro, in the lab, under in model organism, the effect. And that's important because it allows you to understand the scientific mechanism, scientifically the mechanism. But then upscaling and thinking about industrial production requires knowledge, as Marcello has, on technology of production of algae and doing this sort of things. So I've been also grateful to these technological partners to enlarge the possibility of application. And last but not least, the fact why I'm grateful to be invited here and things, because meeting people like Yoko, Didik, Romy, Riza, I mean, it's always beautiful to meet other scientists genuinely interested in theoretical challenges. You know, in the academy, there are also lobbyists that are resistant to change because they have a dominant position, and then if you come with a new idea, it's not necessarily well accepted. What I found here in Indonesia is, also in India I had this feeling, indeed. It was a very nice, open attitude to, how can I say, to discuss. And I don't like to be pleased just to say, oh, your idea is very good. I like questions, I like criticism, and to build together this, I think this is a real good opportunity in this interaction that we are reinforcing now. Thank you.
Thank you, Stefano. I appreciate your remarks. Marcelo, be gentle, be gentle.
First of all, I want to thanks you for our organization, to all for people to coming and to digest and following this new technology, this new way of thinking also. You asked some thoughts. There are a lot of thoughts, of course, because I also collected several reactions, several problems that with this technology we can fight, we can solve. I agree with Stefano about technologies and about type of production, about application in field. And now in this moment it's also quite mature. I'm talking about application of macroalgae with the Noself technology. It's quite mature now, also many thanks to Coppert family, to from the beginning of this industrial pushing, because with Coppert we have also a lot of discussion how to bring this technology in field. So a lot of this maturation in thoughts, in application, cost, type of products, and now also we have also a serious industrial plant in Indonesia. There is I think is a very, very good example of interaction between science, applied science, technology, new technology, and good relationship and discussion. And what mentioned also Tirza before, I like also Romy, good interaction, sometimes also becoming from the field, some interaction, some problem becoming from the farmers, from the industrial point of view, from technology point of view. So also in this case, I think the collaboration between professor, university, whole team, M2M, Coppert, I want to thanks all to and also I want to thanks about this good example of the fertilization and discussion each other. It's yeah, you asked me to be short, but you asked me also to say several thoughts. There are of course several thoughts. What I want to say also, I want to be finished. I fully agree with Professor Mazzoleni. I have a very good feeling, and I noticed the same. We are talking before about this. There is also a very sincere understanding, sincere volontà. It's a sincere type of open mind to new technologies for new challenge, for One Health challenge also. So I conclude. Thank you very much.
Thank you, Marcelo.
If you want, I can continue. I have a lot of the other thoughts.
Oh, thank you very much. Well, I think time has come to call the day to an end, at least this part of it. And I would like to thank those people who I surprised by asking them questions. Thank you very much for responding. If there's one thing I've learned in the course of my career, better speak the way you think, otherwise no use. And I'm very grateful to all of you to have participated in today. I will say one word which is close to my heart, and I know it has nothing much to do with the topic of today, although indirectly it has. That's namely the fact that the world is not looking any better. You know, we're not going to talk about all the recent developments in Europe and Russia and everything else, and the role of the states. That's all there in China, and difficult enough. The consequence will be, gentlemen, ladies, that the pressures on the private sector will expand enormously, and especially in agriculture, more vulnerable sectors because they're not mobile. You know, you're talking about land and labor, and we all know that agriculture labor, yeah, somehow in the history of mankind they always have been lowly paid. It is a fact of life, and we all know that the Bangladeshi are in the Middle East, and the people from Myanmar are doing the oil palm plantations in Malaysia, and you know, the Indonesians were doing the oil palm plantations in Malaysia in the earlier days. They have progressed to other places. This is the reality of today, and I know that it's just a remark like this, and I know that not a single individual person of us can do anything about it, but I'm saying it so that you are also aware that many pressures will come, they will expand. And wise private sector will anticipate it. And also, of course, many people talk about globalization and decoupling, deglobalization. We hear too much about it. In fact, it may be very much so that the need for certification becomes more strong, the need for product safety becomes more strong. Yes, thank you very much, Didi. So that is one remark I want to make, because I'm Taco Botema. And aside from that, I want to thank all of you and everyone, especially Didik and Pa Bungaran, for helping with this get-together. All of your comments we have recorded, and we will find ways to continue the communication as we have started it or restarted it today. So with this, I would like to close our communication, and a final thanks to Riza and to Romy, my brothers in arms on the research. Let's continue to work together and have a good trip back home. Thank you very much.
Thank you very much, Mr. Speakers, Professor Bungaran, and all distinguished participants. I have two requests that all participants would like to take group pictures with the speakers, Professor Bungaran, and to show that and confirm that we did have a very good and fruitful deliberations. And also, quite a few numbers of participants also have requested to have the copy of the presentations. So we will wait for the confirmation from Dr. Riza, Dr. Romy, and Dr.
Wait a moment, and allow me, if you want to have a presentation, kindly send a request to Anne.
Yes.
Okay, that's the way. We will make them available, send a request to Anne.
Yes. I was just going to say, when I sent you email of invitations, so you can use that email and we will respond accordingly. So thank you very much. Let's
take
the group pictures now.
Okay, picture here.